Hello? 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 My monitoring is on. Hello? 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 Lecturing is thirsty work. I had to get a huge glass of water to prepare for this. I knew if I didn't have that, we wouldn't be doing this for like longer than like an hour. Thank you guys. Thank you. I definitely ate way more sushi than I realized it was gonna be. I thought 70 was not going to be as bad. It's not gonna be that bad, but it was pretty bad. I didn't even eat breakfast or lunch to prepare for it, and it still ended up being so much. Backstory for like people that didn't hear. If you didn't know, Hololive has like a collaboration right now with Kura Sushi, which is like a revolving sushi bar. And you have to eat $70 worth of food to get a ticket to be able to get one gear. It's like a hat, I think, but I don't think they even showed off what you get. They just showed a picture of a hat silhouette with a question mark. And it has Ina, Crony, and Gigi Murin like on there. Either way, I knew I wanted it. I don't know what the hat looks like, but I was determined that I'm gonna get it. I'm going to get that. And so I didn't have anyone to go with me because it was kind of last minute. And so I ended up eating all the $70 by myself. And I realized I should have gone in there with like a game plan. And I didn't go in there with a game plan. And I was just eating whatever. And then like by the halfway point, I realized, oh my gosh, I still have like $40 worth of food to get through. And then I started ordering things that were not efficient. Like I ordered takoyaki and it was really heavy and not very tasty. And then I said, okay, I have to be strategic with how to get through the last $35 or so. And yeah, I had to find like there's like some plates that were like, oh, just one leg of snow crab. And I said, oh, that's probably not that bad. It was still really heavy. Oh yeah, I wanted to go for the Pikmin collab, but I was really busy during that time. Oh, I'm jealous that you got to do that. I thought I wouldn't. Honestly, I came into this challenge thinking $70 wouldn't be so bad, but I was surprisingly really full after all of this. Gas station sushi. I don't know about gas station sushi. Though, I know people like bag on grocery store sushi, but sometimes grocery store sushi can be kind of good. I guess it depends on what grocery store you're going to. Maybe like Walmart isn't it? But I've been to like H Mart sushi. H Mart sushi's good. $70 for how full I felt. It was not bad. I did eat like dessert at the end. The dessert was like $7. It's kind of OP. I also cheated because I ordered a soda and then I ordered it again, even though refills are free. And they're like, "Oh, refills are free, right?" And I was like, "Oh, I'm so sorry. I didn't know refills are free. I did know refills are free, but I was like, I need to like get $4 off of my. I need to add $4 onto my tab real fast, and I just want to like soda. I don't want food." Yeah, H Mart is maybe cheating. Oh, it deleted your message. It deleted your message, Caldonia, but I see it. Thank you for the good luck. It was too many exclamation marks. It's okay. Thank you so much. And so it was kind of awkward though, because when I went to the Kura Sushi, there was like not many people eating there alone, but I think there was another guy that was there for the Hololive collab specifically. And I could see what he was doing. I saw he was pacing himself very slowly. Like he ordered a lot of beer and he kept ordering ice cream. He ordered like plate of ice cream. And I was like, Wow, is this a strat? Is this like the ice cream strat? And I don't know if it was a good idea. I feel like the ice cream is kind of big and it's kind of filling and cold compared to like a piece of sushi or maybe two. But I don't know, he like nonchalantly got through it all and then asked for his little ticket. Hey, don't. Oh, it's haha. It's not working because I'm too high up. I didn't pin it to my thing. I need to pin it. I dodged your bonk. I have to. And it felt funny like asking the server at the end for the Hololive ticket redemption. It didn't seem like they really like were that aware that people were gonna go for this thing, at least at my store. And so I like had my receipt and I said, "Hello, I'm here for my gear pass redemption, please. Thank you." And the guy looked at me like, "Huh? What do you want?" Don't worry, Ripco. I am making that. I have to make an animation for it because the ones I like saw that I could just put, they weren't satisfactory for me. So I wanna make my own little animation or you spray me. But don't spray me. Maybe I also want to make one for an autoclave because everyone likes throwing me into the autoclave for some reason. I don't get it. You guys are haters. You guys don't like seeing a bacteria girl succeed. You just want to throw her into the autoclave every day. Anyway, I yapped a lot, but we're reading a textbook today. It's the Microbiology textbook that is available at openstacks.org. This is a open access textbook. Open access textbook by Rice University at OpenStax. Creative Commons Attribution 4.0. Okay, what I learned while I was like looking for things to stream, you need to have this specific license is what you need so you don't get bonked by a stream. If you don't have this, then you don't have actually permission to like stream the text and content. You asked if you could take extra hats. I feel like I should have asked for like maybe another pass. The guy didn't even realize it was like a $70, like you needed to spend $70. He didn't look at my receipt at all. I should have maybe been like, "Oh, can I have like two? One for my friend?" I didn't have a friend there, but you know, I could have lied. But I was just too honest of a bacteria girl. I just said, "Oh, it's for $70 and I spent $70 if you look at my receipt." And then I even had to point in the drawer where they like had the passes stored. I said, "It's that one. It's that one right there with the anime girls. The 3 anime girls, I want that one right there." And then he was just like, "Oh, okay. Whatever." Anyway, this is accessible at openstacks.org. I have to put this here too, just in case, you know, they bonk me for not sharing where this is from. So go to openstacks.org if you ever want to see this. Oh, so the Hololive Gear Pass ticket, I think you get a ticket and you're supposed to bring it in July and you redeem a hat that we don't know what it looks like. We don't know what's on it. We don't know anything about the gear. But I just wanted it because I saw Gigi Murin was on it. On the ticket, at least. Redeem it at Kura Sushi. So you have to go back in a few months later. It's like a double whammy. Like, they want you to spend $70, and presumably, you're gonna come back to spend even more money. Because you're likely... if depending on where you live, you're likely not going to a Kura Sushi just to redeem something. For me though, it's like walkable, so I'll just walk there, hand it in, maybe get sushi, or maybe just go elsewhere because there's a lot of other good restaurants nearby. I don't know if I really care to eat at Kura Sushi again. Yeah, a citation page for a free textbook. Very good. Honestly, I'm not familiar with this textbook, but just doing a look around, like a quick look through, it's really good. And honestly, I've never heard of OpenStax before until I was searching for free textbooks to use on stream, and I was like blown away by what resources you can get for free. And I'm not saying pirate textbooks, but I will say that's kind of what I did in undergrad. I didn't really find it was just way too expensive, man. But it looks like there's so many free resources now. Oh no, you're in the middle of nowhere for no Hololive Kura sushi. I think it's kind of crazy. It's pretty last minute. You have to go to this restaurant from the 16th to the 22nd, so this weekend only, and then go back later in July for the redemption. It was really short notice. Uh, and I was asking people if they wanted to come with me. My treat. It was just to make it easier, but it's kind of hard to find somebody to go to sushi last minute this weekend. So I had to bear the brunt of eating $70 worth of sushi. Such a terrible burden for myself. But it was worth it. Yeah, I'm not saying to pirate textbooks, but, I'm just saying, maybe I never got bonked. Maybe I never did anything wrong. Maybe it's okay, but I didn't say that. Get stuffed on sushi again for an extra ticket? Maybe I don't know if I'm gonna be. I'm not sure if I'm gonna be ready to do $70 worth of sushi again. $250 for a new textbook. Textbook prices are another aspect of science that but it's just I guess it's not really science. Academia in general, it's just insane. In undergrad, the way they would make you buy a textbook and then you're forced to get it because there was a homework component to it was absolutely bonkers. The online you have to, what is it, McGraw-Hill online homework programs. Oh, it was terrible. So you couldn't even get a used copy because you needed to buy a new copy, or else they wouldn't come with the homework program to submit your work. I feel like stuff like that should just be illegal. Absolutely shameful. Shameful practices. So I love things like this. All that work and you end up with a job that doesn't even use your degree. It's just so sad. The price of to get a degree is just way too high. Considering it's not even a guarantee to be able to use what you get. Or what you study, I should say. Yeah, I love professors like that. I had so many professors that said, hey, I don't want you to buy a new textbook. A new textbook is stupid. They're charging you, they charge so much money for these textbooks that you don't really end up using that often. Or when professors say you need to use this specific version of the textbook, and there's barely any differences between the most recent up-to-date version and the version from a decade ago. Just such a joke. I really appreciated professors that would say, I'm copying the things you need from the textbooks, and they will be on our our platform where I disperse assignments and reading and all of that. It was just so much more kinder to the students. Oh, the professors that are part of the textbook pyramid scheme. Oh, those are the worst. They really push you to get make sure you have the latest up-to-date copy. You have to make sure you have version 13, the 13th edition, or else it's not gonna be good enough. Give me a break, professor. I know who you are. I know what you are. You're a royalty. You're getting royalties. So yes, this OpenStax textbook, it's free, peer-reviewed, openly licensed, and apparently some colleges, college courses actually use these in their, like, as the textbook for their course, which I think is so cool. Go Rice University. I would have appreciated if this was something I had, like, I knew about or had in undergrad. So many philanthropic support. I'm just looking to see. Hmm. Yeah. Yeah, I can imagine things like this are probably really expensive. It's probably why it takes all this kind of like money to be donated. Which sucks that it's so expensive. Study where you want, what you want, when you want. There's no one like I could say like I know too much about. I'm like surprised Chegg is on here. I know Chegg as like the website where you get like homework help that you have to pay for. So I'm just like wondering, they donated to this? There's also a lot of like computer like companies here, like HP, Intel. It's like a mix. Though I'm gonna guess it's not. Obviously it's not specific to the microbiology textbook. It's just the OpenStax in general. So there's probably I get it. I think open source like material tends to be way more popular in like computer science technological things. I find that is still lagging in the biological science space. People are still very much, it's hard to convince scientists that it's okay to like let your science be available to read to many people for free. Yeah, that's what I figured, like from programming. Study where you want, what you want, when you want. It's the most low. I don't know if you can tell. I guess you can on OBS. It's like the most low res advertisement. It's like so fuzzy. This is very much giving graphic design is my passion. Let's see what chapters we have. An invisible world. What our ancestors knew. Types of microorganisms. Oh, there's even review questions. We can study and learn, guys. How we see the invisible world. The cell. Prokaryotic diversity. The eukaryotes of microbiology. Acellular pathogens. Microbial biochemistry. Whoa. This was a part of biochemistry I really liked when I went through undergrad. And I wish there was like more of it when I went through my courses, but this was like such a small niche. Microbial metabolism. Oh boy. Microbial growth. Biochemistry of the genome. Okay. I really suck at like genetics. I think my knowledge on genetics is pretty poor, despite being a cancer. Like I study a lot of cancer biology. I feel like I'm just not very like. I never really studied a lot about DNA and genomics and all that. Where's my gram stain? What part of the ribosome does this antibiotic target? Control of microbial growth. Maybe we will ignore this chapter. Maybe this chapter we don't have to look through this. This chapter, no, we don't need to know how to stop the microbes from growing, guys. Okay, maybe we don't also look at antimicrobial drugs. We're gonna skip chapters 13 and 14. Disease and epidemiology, microbial mechanisms of pathogenicity. Innate non-specific host defenses, adaptive specific host defenses, diseases of the immune system, laboratory analysis of the immune response. It's kind of impressive how in-depth this goes. because there's a lot of like even immunology in here, and I know it's like hard to not have immunology when you're talking about microbiology. I'm just impressed. I never took a proper microbiology course because it wasn't required in my for my degree. This one's very specific. Skin and eye infections. Respiratory system infections, urogenital system infections, digestive system infections, circulatory and lymphatic system infections, nervous system infections. It's a lot of it. It's pretty interesting how deep it goes into certain infections. We're gonna learn so much, you guys. Honestly, this is pretty useful for me too. I just kind of want to learn. Blank, blank. I'm skipping all the preface. Sorry, guys. Actually, let's look at some pictures. Our art program is designed to enhance students' understanding of concepts through clear and effective illustrations. Oh, cool. They have an art program? That would be one thing I also want to stream. I want to stream, like... I wanted to be a science illustrator at one point, so a science illustrator usually are the ones that do diagrams like these that you see in textbooks and papers. But it's a really specific kind of art program you have to go through. And I know a lot of them even go, there's like a master's program specific for scientific illustration. And I realized I don't know if I wanted to go down that path. I think it sounded more fun. It sounded more interesting in my head than it actually would be. Yeah, I remember I sat in on a class for scientific illustrators because I was just curious what it's like. And it's a lot of work. A lot of times what I saw in the course that I sat in on was you basically have to get in-depth, like really in-depth knowledge about the specific project you're going to work on. And so you have to know, okay, I need to diagram all these things. They were honestly really talented artists. They had to be experienced with so many things, like also animation. I think nowadays a lot of the people that are hiring scientific illustrators, they also want to see animations of these things. So like this thing moving in the body or whatever. It's pretty complicated. Also, unfortunately, it does not pay well. And that was that was kind of also another reason I realized that I don't think I would want to do that as a full-time living. It's not a really well-paying job. Despite how educated you have to be. You have to be pretty educated and skilled. I mean, science and art aren't exactly, they don't tend to overlap that much. Flagella got a flutter. Yeah, exactly. And another thing that I think is a little worrying about scientific illustration as a job is there's so many things like biorender nowadays, and also just AI image generation. It feels like that field is getting targeted really. They're getting ousted. Which sucks, because I don't think AI is even close, as we've seen with like the giant rat testicle AI generated figure. But biorender is like pretty good. I think it's like good enough for people to not want to pay an artist. Because an artist is kind of expensive. I drew an abstract figure for somebody and for my lab, and they paid me for it, which was very generous. And I got paid a decent amount, but then it made me think, wow, like, I feel like that expense is kind of a lot just to submit like a paper. So I could see why people don't. They hate graphical abstracts. So who wrote this? Nina Parker, Shenandoah University. PS and MS from University of Michigan, and her PhD in immunology from Ohio. Ohio. She joined Shenandoah University's Department of Biology in 1995. 1995. Man. Not the AI graphics. We can't keep talking. We can't keep using the AI graphics for science. It's so bad. But that's not gonna stop people. It's not lately. I've seen a lot of AI graphics generated for science, and they have not. Fortunately, I don't know if maybe unfortunately have not been as bad as big rat testicle picture, but they are definitely getting better. And of course, that like getting better could mean bad things or good things depending on how you feel about AI. Dr. Mark A. Schneegert, Wichita State University. He was a postdoctoral fellow at Eli Lilly. Did you guys see how much Eli Lilly is like going how crazy they're going stock wise lately after they dropped the content nuke of we have Ozempic in tablet form. Like they're like, no more. We're skipping injectable Ozempic. Now you can just take this in a daily pill, and their stocks went crazy. The whole weight loss pharmaceutical development is like such an interesting thing. It's crazy that I think it's like the biggest money generator. Everyone just wants to lose weight. And also now in pill form, I think people were pretty apprehensive about taking Ozempic because of the fact that it's injectable. I think injectables are always going to be scary for your average person, but you tell them they can take a pill every day and maybe lose like 15 pounds, and people are gonna they go insane for it. Yeah, it's pretty expensive though. I think Ozempic as an injectable, it's weekly, and it's like hundreds of dollars. Yeah, I mean, globally, obesity is on such a huge rise. It's not even just like, I know some people will say it's an American problem, but it's just happening all over the world. You're trying to gain, not lose. You know, I know a trick to gain weight. You can go get $70 of sushi at a revolving sushi bar. That's a easy way to gain weight. Caledonia. $700 is pretty, yeah. It's pretty insane. I don't know how much this pill form is gonna take. It's like gonna cost, but since it's daily, I can't imagine that the price is gonna be that different. It might be even more expensive. I'm sure we'll see prices go down whenever other companies start doing this daily pill. Yeah. Yes, per month. I think I'd love to go over more about Ozempic just because I think it's a fascinating therapeutic. If you like guys haven't seen, they keep like finding like how it's affecting so many different mechanisms, and they go, "Oh, it's actually helping you like fight addiction. It's helping you fight like you can quit smoking if you take Ozempic. Oh, it's really easy to stop like doing drugs if you take Ozempic." And it's also things like it helps you not age and helps you live longer. They just keep calling it like the magic therapeutic for everything. Oh, I didn't know that about the origin. Interesting. Yeah. Oh, a deep dive on Ozempic sounds like it would be fascinating to do. And there's also like the issue of Ozempic is that it's also it's a therapeutic for diabetes, at least initially. And it's hard to it's like difficult because there's so many supply issues with the therapeutic because there's so many people that are taking it just for losing weight. But it's not as like life threatening as the people who need it for their diabetes management. It's rough. And there's also like issues with people who aren't necessarily in an unhealthy weight range being prescribed it still by doctors online. It's crazy. You can just go to a website. I'm not going to say like how to do it. I don't recommend you doing this if you're listening. You could just go to a website and like get like some online doctor to prescribe it to you so easily without ever getting checked up on. It's pretty insane. I don't know if it should be like that, but I also think it's hard to like it's just hard to like fix that problem. Who else? Dr. An Su Tu, born in Saigon, Vietnam. I'm just trying to see if I recognize any specific scientists here. Saskatchewan. Or do I recognize any universities? Like, not that I don't recognize these universities. Are there any universities that I am connected to? Not that I really see. The Mississippi University for Women. It's really scientists from all over the place. It's pretty nice. What a like broad science field. Overall, it's super effective at weight loss and darn good. Yeah, A1C. Yeah, that's also another thing. Like, if this is like so effective for weight loss, I do like that there's this option that is not as invasive as like gastric bypass. I don't know about the efficacy, like with people that are like dangerously obese. I always hear like you can lose up to like 15 pounds or so. But I'm guessing at that point, maybe it's not 15 pounds. Surely they would need to lose more than 15 pounds. Look, oh my gosh, the sea turtle's covered in oil! A veterinarian gets ready to clean a sea turtle covered in oil following the Deepwater Horizon oil spill in the Gulf of Mexico in 2010. I remember this! After the spill, the population of a naturally occurring oil-eating marine bacterium called... Oh boy, this is going to be me butchering a bunch of bacteria names. I'm sorry to my fellow microbes. Alcanivorix vorcumensis skyrocketed, helping to get rid of the oil. Scientists are working on ways to genetically engineer this bacterium to be more efficient in cleaning up future spills. Oh, a bacterium that eats the oil. That's fascinating. So sad. This poor turtle. Don't worry, guys. The oil-eating bacterium are here to save us. Okay, it's time to understand the invisible world. This is the world I'm from, guys. This is my world. I'm sharing my world with you all. From boiling thermal hot springs to deep beneath the Antarctic ice, microorganisms can be found almost everywhere on Earth in great quantities. Microorganisms, or microbes as they are also called, that's me. That's me, guys. That's me. Are small organisms. Most are so small that they cannot be seen without a microscope. Guys, you can see me. You can see me. I'm not that small. You can see me. Look, I'm big too. Okay, I'm going back to being small. What if I'm so small you can't see me? They want me to be this small. They want me to be this small so bad. Okay, let me get big. And where'd my textbook go? Okay, I lost my page. Most microorganisms are harmless to humans, and in fact, many are helpful. They play fundamental roles in ecosystems everywhere on Earth, forming the backbone of many food webs. Many use them to make biofuels, medicines, and even food. Without microbes, there would be no bread, cheese, or beer. That's true. Kind of crazy how many processes in our like day-to-day life a bacteria affect. I forget. Yeah, it's just kind of insane. Can you imagine no bread, no cheese? I could do without beer. I don't really care for alcohol to begin with, so you know. But no bread and no cheese? Okay, I can also do without some cheese because I can't eat a lot of cheese. But no bread? No bread? Our bodies are also filled with microbes, and our skin alone is home to trillions of them. Trillions and trillions and trillions. I like the name of this paper they cite. A jungle in there. Bacteria in belly buttons are highly diverse but predictable. It almost sounds like a sounds like a own. Like they're highly diverse but they're predictable. Also, why is it specifically belly buttons? I can't say I'm a fan of the belly button. I just don't like anything there. Kind of a weird area on the human body. Although much more is known today about microbial life than ever before, the vast majority of this invisible world remains unexplored. Microbiologists continue to identify new ways that microbes benefit and threaten humans. No, I'm not threatening you guys. Some microbes may be threatening you guys, but I'm not threatening you guys. I'm benefit. I am the benefiting you guys. Benefit. What our ancestors knew. By the end of this section, you will be able to describe how ancestors improved food with the use of invisible microbes. I feel like the mentioning of invisible microbes, I don't know how many times it needs to be said. Describe how the cause of sickness and disease were explained in ancient times prior to the invention of the microscope. Describe key historical events associated with the birth of microbiology. The birth of microbiology. Oh man, I'm always, when I would read these textbooks as a student, and they would have like the little squares saying, here's like a clinical example, I would just go like, yeah, I don't care. I'll read it. Because it's relevant and we should know why these are important. Clinical focus. Cora, a 41-year-old lawyer and mother of two, has recently been experiencing severe headaches, a high fever, and a stiff neck. Poor Cora. Her husband, who has accompanied Cora to see a doctor, reports that Cora also seems confused at times and unusually drowsy. Based on these symptoms, the doctor suspects that Cora may have meningitis, a potentially life-threatening infection of the tissue that surrounds the brain and spinal cord. Well, I guess that would make sense if she says she has a stiff neck and she's confused. I feel like that also to me jumped as that's like the equivalent of me jumping on WebMD and going, my head hurt and my neck stiff, and then they say I like have the worst thing imaginable. Meningitis has several potential causes that can be brought on by bacteria, fungi, viruses, or even a reaction to medication or exposure to heavy metals. Although people with viral meningitis usually heal on their own, bacterial and fungal meningitis are quite serious and require treatment. Cora's doctor orders a lumbar puncture spinal tap to take three samples of cerebral spinal fluid from around the spinal cord. And there she is. It's like The Substance. Did you guys watch that movie? They have to take it right from the spinal cord. A lumbar puncture is used to take a sample of a patient's CSF for testing. A needle is inserted between two vertebrae on the lower back, called the lumbar region. CFS should be clear as in the sample. Abnormally cloudy CFS may indicate an infection but must be treated further to confirm the presence of microorganisms. Ooh. That's crazy. Can you, oh my gosh, I feel like that's so painful. I can't imagine that. The samples will be sent to laboratories in 3 different departments for testing: clinical chemistry, microbiology, and hematology. The samples will first be visually examined to determine whether the CSF is abnormally colored or cloudy. Then the CSF will be examined under a microscope to see if it contains a normal number of red and white blood cells and to check for any abnormal cell types. In the microbiology lab, the specimen will be centrifuged to concentrate any cells in a sediment. Hey, basic lab techniques. I know this stuff. I know how to do this. Put me in, coach. I can centrifuge and concentrate into a sediment. This sediment will be smeared on a slide and stained with a gram stain. Gram staining is a procedure used to differentiate between two different types of bacteria, gram-positive and gram-negative. About 80% of patients with bacterial meningitis will show bacteria in their CSF with a gram stain. Cora's gram stain did not show any bacteria. Gasp! What a twist! It's not bacterial meningitis. But her doctor decides to prescribe her antibiotics just in case. Part of the CSF sample will be cultured, put in special dishes to see if bacteria or fungi will grow. It takes some time for most microorganisms to reproduce in sufficient quantities to be detected and analyzed. What types of microorganisms will be killed by antibiotic treatments? What will be killed? Me! I'll be killed. JK, I'm antibiotic resistant. You can't get me like this. Most people today, even those who know very little about microbiology, are familiar with the concept of microbes or germs and their role in human health. Okay, I'm gonna pause right there. I feel like I've been the bacteria Vtuber girl for actively for about 2 months, month and 2 months now, and I've walked into some communities that don't know much about science. I'll have you know, I don't think they're very familiar with microbes at all. I've had to explain what a microbe is each time. And also, I think I've noticed there doesn't seem to be a connection of what a germ is and how it's a microbe. Like, germ is just like this a concept that is very separate than anything like bacterial or microorganism. It's just a germ. At least that was my experience. Maybe I'm walking into some low bar communities that don't know much. School children learn about bacteria, viruses, and other microorganisms, and many even view specimens under a microscope. Okay, pause again. I refute this. I did not learn about anything about bacteria or viruses, and I especially did not have a microscope in my school days. Don't know who they're talking to. That's not me. But a few hundred years ago, before the invention of the microscope, the existence of many types of microbes was impossible to prove. By definition, microorganisms, or microbes, are very small organisms. Many types of microbes are too small to see without a microscope, although some parasites and fungi are visible to the naked eye. Like me. They didn't mention me, but I'm visible to the naked eye. You guys see me? You guys see me right here? Do I need to get bigger again? No, you guys can see me. Humans have been living with and using microorganisms for much longer than they have been able to see them. Historical evidence suggests that humans have had some notion of microbial life since prehistoric times and have used that knowledge to develop foods as well as prevent and treat disease. Wow. In this section, we'll explore some of the historical applications of microbiology as well as the very early beginnings of microbiology as a science. Wow. Oh, fermented foods and beverages. I love this topic. I want to do some YouTube videos on bacteria used in food. But I gotta actually record content, and recording content is so hard sometimes. People across the world have enjoyed fermented foods. Oh wait, hold on. I can't see something. Okay. People across the world have enjoyed fermented foods and beverages like beer, wine, bread, yogurt, cheese, and pickled vegetables for all of recorded history. Any pickled vegetables fans? I love a good pickled vegetable. Kimchi, pickles, pickled like any vegetable, like a quick pickle. A quick pickle is really, really good. If you like Japanese cuisine, you probably really like pickled vegetables, unless you're the type of person to like Japanese cuisine that's not actually Japanese cuisine and it's like the Western fried foods. Then I guess you're not part of that. I feel the people that don't like a good pickled vegetable, I can't trust them. Can't trust you guys. There's just no way you're not gonna enjoy a pickle. There's just no way you're telling me, like, a lightly pickled vegetable, it doesn't even have to be super pickled, like, a light, like, one tossed in vinegar and some salt for just even, like, 10 minutes, you're not gonna tell me you don't enjoy that, right? No way. No way. Discoveries from several archaeological sites suggest that even prehistoric people took advantage of fermentation to preserve and enhance the taste of food. Archaeologists studying pottery jars from Neolithic village in China found that people were making a fermented beverage from rice, honey, and fruit as early as 7000 BC. Wow, that's kind of insane. How do you even like go about that? I guess you're just putting stuff together. It's kind of just like, yeah, GG, like honey fruit, that tastes good. Maybe we throw a little rice in there. Maybe we forget about this like thing we put together for like a few months. Maybe I take a sip, take a little sippy sip because it smells good still after a few months. Braver than me, those people. Braver than me. I would not suggest taking a sippy sip of something that you don't remember putting together. Production of these foods and beverages requires microbial fermentation, a process that uses bacteria, mold, or yeast to convert sugars, carbohydrates, to alcohol, gases, and organic acids. While it is likely that people first learned about fermentation by accident, perhaps by drinking old milk that had curdled, or old grape juice that had fermented. That's also real gross. It's just so gross to think about. Like, oopsie, just drank some old juice. Oh, it's kind of tasty. They later learned to harness the power of fermentation to make products like bread, cheese, and wine. The yeast fermentation. Look, a little yeast into bubbling starter into bread. That doesn't. I'm gonna be honest. That bread's not looking that good. It's kind of looking like it's okay, but I feel like it's not that good looking. That doesn't look very That doesn't read very sourdough. Maybe it's not sourdough though, and I'm just like projecting. It just looks a little yellow and cakey to me. Anyone else see that? I'm sure it's really tasty bread. Let me not hate on this baker, Mr. Dre Belbis at Flickr. Sorry for making fun of your bread. A microscopic view of Saccharomyces cerevisiae. I'm so bad at pronouncing the name of yeast. They're responsible for making bread rise. Left. Yeast is a microorganism. Its cells to metabolize the carbohydrates in flour and produce carbon dioxide, which causes the bread to rise. The Iceman, and evidence of early treatments. Prehistoric humans had a very limited understanding of the causes of disease, and various cultures developed different beliefs and explanations. While many believed that illness was punishment for angering the gods or was simply the result of fate, archaeological evidence suggests the prehistoric people attempted to treat illnesses and infections. Okay, simply the result of fate part, I think there's still a lot of people that still believe in this. I still feel like there's a lot of people that still think they got some kind of illness because of fate. I don't think that's that uncommon nowadays, unfortunately. One example of this is Otzi the Iceman, a 5,300-year-old mummy found frozen in the ice of the Otzi Alps on the Austrian-Italian border in 1991. Because Otzi was so well preserved by the ice, researchers discovered that he was infected with the eggs of the parasite Trichursis trichuria, which may have caused him to have abdominal pain and anemia. Oh no, poor Iceman. Poor dude. Researchers also found evidence of Borrelia burgdorferi, a bacterium that causes Lyme disease. Some researchers think Otzi may have been trying to treat his infections with the woody fruit of the Fomitopsis botulinus. fungus, which was discovered tied to his belongings. This fungus has both laxative and antibiotic properties. Was also covered in tattoos that were made by cutting incisions to his skin, filling them with herbs, and then burning the herbs. Oh, ow! Ugh. Like, I get it. If that's the medicine that you've got going for you, it makes sense. But oh, that sounds incredibly unpleasant. That sounds very, very unpleasant. I don't like it. Several. I just can't imagine, like, you cut your skin, you put herbs in it, and then you burn it. Oh, that sounds so painful. Ugh. God bless modern medicine. Early notions of disease, contagion, and containment. Several ancient civilizations appear to have had some understanding that disease could be transmitted by things they could not see. This is especially evident in historical attempts to contain the spread of disease. For example, the Bible refers to the practice of quarantining people with leprosy and other diseases, suggesting that people understood that diseases could be communicable. I guess that's true. They did know that you can transmit diseases. Ironically, while leprosy is communicable, it is also a disease that progresses slowly. This means that people were likely quarantined after they had already spread the disease to others. Yeah, medical treatments, it's pretty insane. I'm sure, like, this section, we're going to see some pretty scary things. I mean, you don't even have to go that far back to see some insane treatments. Like, we can talk so much about things, involving what they did to treat, like, mental illnesses. It's pretty sad, pretty nasty. It's like we've come a long way since then, though it feels like maybe we might be taking a few more step backs than we would like to see. Ironically, while leprosy is communicable, it is also a disease that progresses slowly. This means that people are likely quarantined after they had already spread disease to others. Clean water and sanitation are among the most important elements of healthy societies. The earliest examples of urban sanitation systems are from the ancient Indus Valley civilization cities of Mohenjo-daro and Harappa, located in current-day Pakistan. Oh, interesting. Constructed in 2500 BCE, about 4,500 years ago, the cities had complex network of wells, baths, and drainage systems that stored fresh water and carried waste away. Good for them. Good for them. No poo-poo doo-doo near all over their country. There's a Civil War fort kinda near me. Tweezers, bone saws, hammer, and a bottle of liquor. Oh my gosh. I have heard of things like that where they would just drink. They were drinking to get through, like, because they needed it to get through the surgery. That's so insane. That's so crazy. Oh my gosh. Well, good thing we don't deal with that now. Maybe some pain relief, just maybe. About 2,000 years later, people in the ancient Greek civilization attributed disease to bad air. Malaria, which they called miasmatic odors. Not wrong. Yeah, I mean, hand washing in general, it's relatively modern, which I feel, when I say that, it sounds like an insane person thing to say, that hand washing is modern. We've made so many developments in, like, just a few decades compared to, like, all of history. It's pretty nutso to think about. In Rome, they built aqueducts, which brought fresh water into the city and a giant sewer, the Cloaca Maxima. The Cloaca Maxima. That was the name of their sewer? The Cloaca Maxima, which carried waste away and into the river Tiber. Okay, some researchers believe that this infrastructure helped protect the Romans from the epidemics of waterborne illness. Yeah, it's not ancient Rome. Nope, nope. Oh my gosh, not yeah, that the big hole on the bottom of the skull is also another insane thing. Okay, we have an ad break coming up and I'm going to just look at this picture while the ad break is going. Yeah. I gotta drink more water while I'm doing this. I'm like forgetting to drink water and hydrate myself. It's like I'm talking a whole lot. I need to be hydrated. My throat needs to be prepared to yap and yap about these papers. The cloaca maxima. Thank you for the hydration. The cloaca maxima, the greatest sewer. I like that. The greatest sewer ran through ancient Rome. It was an engineering marvel that carried waste away from the city and into the river Tiber. These ancient these ancient latrines emptied into the cloaca maxima. Something about that caption is so funny to me. It's so this was the cloaca maxima shown in red. This I see. So all of this red line, it's hard to see. And then where's the river? Where is the river? Oh, down there. Their latrines are kind of funny. I've never seen Roman latrines like this. So you just like sat in this public area with like strangers and your homies and just just wasted on these stones. The design's not even that bad. I see how we got to the toilet just looking at this. Like that's kind of toilet like lid shaped. Don't I'm trying to figure out what's going on here in these like holes. What were we doing here? Did we stick something in there? Or is this just like to make sure you're catching everything? You didn't like miss anything? Also, I'm just like it's like amazing to think they just made these out in the open, like not a single amount of privacy. And it's not like they probably just didn't even think about that. More like they didn't even care. They're just like, yeah, I'm just I'm just dropping one right here right now, buddy. You can keep talking to me while I'm doing that. And then you could just have other people right here too. You could just have a whole like bench of you and your friends just relieving the waste altogether. No walls, no barriers between you guys. It's like kind of beautiful in a way. Why did we develop walls? Maybe we need to bring back this. Maybe society would be in a better place if we all just sat together in our public waste square, and then we sent it to the cloaca maxima, which emptied into the river Tiber. By the way, even before the invention of the microscope, some doctors, philosophers, and scientists made great strides in understanding the invisible forces, what we now know as microbes, that can cause infection, disease, and death. Okay, I don't appreciate the slander of microbes. Again, I have to defend myself every single time. I'm oh I'm good. I'm a good microbe. I'm just here to infect people non-pathogenically. I'm just here to spread my word. Anyway, ad break is over. Welcome back to the people that were in the ad. All we did was talk about the public restrooms during the ad. The Greek physician Hippocrates, 460 to 370 BC. What a year. What a birth year. Is now considered the father of Western medicine. Unlike many of his ancestors and contemporaries, he dismissed the idea that disease was caused by supernatural forces. Instead, he posited that diseases had natural causes from within patients or their environments. Wow. It's kind of crazy to think how could someone know how could someone know all that back then? What a genius. I couldn't even imagine I just could I would be busy thinking about when am I gonna go use the public latrine for the cloaca maxima. Instead, this guy is like thinking about how diseases might be coming from inside the house. Hippocrates and his heirs are believed to have written the Hippocratic Corpus, a collection of texts that make up some of the oldest surviving medical books. Hippocrates is also credited as the author of the Hippocratic Oath, taken by new physicians to pledge their dedication to diagnosing and treating patients without causing harm. Good for them. Good for them, guys. While Hippocrates is considered the father of Western medicine, the Greek philosopher and historian, I know how to say this guy's name. I've searched it before, and I'm gonna embarrass myself. His name's now Thu. Is considered the father of scientific history because he advocated for evidence-based analysis of cause and effect reasoning. Damn, what a genius! What do you mean advocating for evidence-based analysis and cause and effect in 460 BC? He's insane mode. He's just so smart. Again, me in 395 BC. Still hanging out with my friends on the public latrine, probably. Among his most important contributions are his observations regarding the Athenian plague that killed 1/3 of the population in Athens between 430 and 410 BC. See, I probably would have been killed by this plague because I was hanging out so much in the public latrine square and getting diseases. Having survived the epidemic himself, he made the important observation that survivors did not get reinfected with the disease, even when taking care of actively sick people. This observation shows an early understanding on the concept of immunity. Wow. He's just so smart. I just can't imagine seeing those kind of patterns. Because it is just a pattern when you think about it. He just saw these people, they're not getting sick again. And I bet it's because, they were sick before. Marcus Terentius Varro, what a name, was a prolific Roman writer who was one of the first people to propose the concept that things we cannot see, what we now call microorganisms, can cause disease. Can you imagine being the guy who says there's things we can't see causing disease? You probably sound so insane to everyone. They probably think you're crazy. They probably think you're stupid also. In Res Rusticae on farming, published in 36 BC, he said that precautions must also be taken in neighborhood swamps because certain minute creatures, Animalia minuta, grow there, which cannot be seen by the eye, which float in the air and enter the body through the mouth and nose and there cause serious diseases. He's so smart. How did he know? But again, I guess this is just you observe. Isn't that all they did back in the day? Especially Romans and Greek philosophers. They just observed. They were just looking. They just kept looking with their eyes and seeing patterns and saying, I go to these swamps and I get sick. Crazy. Centuries later, Islamic scholars, mainly in Persia, built on the knowledge from Hippocrates, as well as from Chinese and Indian medicine. Foremost among them was Abu Bakr al-Razi. I'm sorry to all the names I'm butchering. I'm sorry. I'm so sorry. Usually referred to al-Razi, or by his Latinized name, Rhazes. What? How did we get there? Name's hard. Especially I'm bad at bacteria names. I'm bad at people's names. It's just so hard. He developed a range of experimental methods to test various aspects of medicine. For example, to select a location for a hospital, Al-Razi hung raw meat around the city and located the facility in the places where the meat took the longest to rot. Whoa. This guy's a genius. He just said, yeah, we're kind of like meat. And he's right. We are meat. And just hung stuff up and said, okay, bet it's gonna take this long to rot. That's how bad this area is then. Wow. Actual genius mode. He was also the first to distinguish measles and smallpox and undertook experiments to determine which available treatments were most effective for each. While Al-Razi did not have the technology to understand the role of microbes, his efforts to identify the causes of illnesses rather than solely focusing on the symptoms were highly influential. Yeah. I mean, I feel like he did so much good work there, just kind of going like, yeah, we know what's going on, but I don't really know what's going on. Kind of impressive. Islamic physicians created one of the most important medical texts in history, the Canon of Medicine. Compiled in 1025 by physician and philosopher Ibn Sina, also referred to as Avicenna. Oh man, they give me like 2 different names to say and I can't pronounce either of them. The encyclopedia included detailed descriptions of the parts of the body, a detailed account of over 800 medicinal substances, and pathologies of several illnesses organized by body part or health event. Wow! I feel like documenting all this information must have been so hard back then. I'm sure this guy, maybe he didn't, I don't know if he did all this, he just probably compiled the work. It's just trying to get information must have been so hard. It's so hard nowadays to get even anything, and we have Google. In the work, Ibn Sina described mechanisms of contagion, indicated that organisms could be infected by foreign substances, and that illness could be transmitted by breath. He said, don't breathe on people. There's things in there. He is also credited with advancing the practice of isolating people who are sick, laying the foundation for historical and contemporary quarantine methods. The canon was translated into many languages and was a primary resource for teaching medicine around the world through the Renaissance and beyond. Wow. Hippocrates, the father of Western medicine, believed that diseases had natural and not supernatural causes. Absolute goat. The historian Thu observed that survivors of the Athenian plague were subsequently immune to the infection. Marcus Terentius Varro proposed that disease could be caused by certain minute creatures. What a nice picture of the statue. I'm not going to check your understanding, guys. The birth of microbiology. Oh, we're getting into like people, like closer to modern times, not just like historians from Greek. While the ancients may have suspected the existence of invisible minute creatures, it wasn't until the invention of the microscope that their existence was definitely confirmed. While it is unclear who exactly invented the microscope, a Dutch cloth merchant named Antony van Leeuwenhoek. I can't. European names are so hard. 1632 to 1723 was the first to develop a lens powerful enough to view microbes. Wow. How'd he do that? Hello, does anyone know how he developed a lens that powerful in the 1600s? In 1675, using a simple but powerful microscope, Leeuwenhoek was able to observe single-celled organisms, which he described as animalcules, or wee little beasties swimming in a drop of rainwater. Why didn't we go with that name? Why didn't we go with that name for like our current history? I want to be called a wee little beastie, not a. I don't want to be a microbe. Wee little beastie. Wee little beasties is so cute. From his drawing of these little organisms, we now know that he was looking at bacteria and protists. We will explore his contributions to microscopy further and how we see the invisible world. Nearly 200 years after Van Leeuwenhoek got his first glimpse of microbes, the golden age of microbiology spawned a host of new discoveries between 1857 and 1914. Two famous microbiologists, Louis Pasteur and Robert Koch, were all especially active in advancing our understanding of the unseen world of microbes. Pasteur, a French chemist, showed that individual microbial stains had unique properties and demonstrated that fermentation is caused by microorganisms. These are now, like, we're getting into history that I'm pretty familiar with. Like my microbiology class never got far, like, later than this. I mean, not, or earlier than this, I should say. I wasn't super familiar with, like, all the Greek and Roman stuff, aside from, like, a passing, like, mention. Obviously, I knew that they did contribute a lot. He also invented pasteurization, a process used to kill microorganisms responsible for spoilage, and developed vaccines for the treatment of disease, including rabies in animals and humans. Wow, go dude. Koch, a German physician, was the first to demonstrate the connection between a single isolated microbe and a known human disease. For example, he discovered the bacteria that can cause anthrax, Bacillus anthracis, cholera, Vibrio cholera, and tuberculosis, Mycobacterium tuberculosis. We will discuss these famous microbiologists and others in later chapters. What a sick picture. Louis Pasteur. And then there's Robert Koch. I like this painting a lot. This painting looks sick. This picture is pretty whatever. As microbiology was developed, it has allowed the broader discipline of biology to grow and flourish in previously unimagined ways. Much of what we know about human cells comes from our understanding of microbes, and many of the tools we use today to study cells and their genetics derive from work with microbes. Okay, I'm not checking your understanding. If you're not understanding, you're not, I don't know what to tell you. Microbiology toolbox. Because individual microbes are generally too small to be seen with the naked eye, the science of microbiology is dependent on technology that can artificially... I'm glad you understand. Good, you understand. That can artificially enhance... that can artificially enhance the capacity of our natural senses of perception. Early microbiologists like Pasteur and Koch had fewer tools at their disposal than are found in modern laboratories, making their discoveries and innovations that much more impressive. Later chapters of this text will explore many applications of technology in depth, but for now, here's a brief overview of some of the fundamental tools of the microbiology lab. Microscopes. They produce magnified images of microorganisms, human cells, and tissues, and many types of specimens too small to be observed with the naked eye. Stains and dyes are used to add color to microbes so they can be better observed under a microscope. Some dyes can be used on living microscopes where others require that the specimens be fixed with chemicals or heat before staining. Some stains only work on certain types of microbes because of differences in their cellular chemical composition. You guys are all learning all about everything I have. This is you need to understand these things in order to, to understand me. Growth media are used to grow microorganisms in a lab setting. Some media are liquids, others are more solid or gel-like. A growth medium provides nutrients, including water, various salts, a source of carbon like glucose, and a source of nitrogen and amino acids like yeast extract, so microorganisms can grow and reproduce. Ingredients in a growth medium can be modified to grow unique types of microorganisms. You need to feed me this. I need you guys to feed me this every day. A petri dish is a flat-lidded dish that is typically 10 to 11 centimeters in diameter and 1 to 1.5 centimeters high. Petri dishes are made out of either plastic or glass and are used to hold growth media. You guys live here. The bacteria buds, we're all in here. We're in the petri dish all together. Test tubes are cylindrical plastic or glass tubes with round bottoms and open tops. They can be used to grow microbes in broth or semi-solid solid growth media. A Bunsen burner is a metal apparatus that creates a flame that can be used to sterilize pieces of equipment. A rubber tube carries gas to the burner. In many labs, Bunsen burners are being phased out in favor of infrared micro incinerators. Is that true? Maybe I don't work in a I didn't work in many labs that actually use Bunsen burners or like just open flames in general. And like we didn't do a lot of culturing. I was much more of a mouse. I was a mouse girly. Are there? Is it micro incinerators? Is this true? Any microbiologists? Any knowers? It would make sense. An inoculation loop is a handheld tool that ends in a small wired loop. The loop can be used to streak microorganisms on an agar in a petri dish or to transfer them from one test tube to another. Before each use, the inoculation loop must be sterilized so cultures do not become contaminated. This petri dish filled with agar streaked with Legionella, the bacterium responsible for causing Legionnaire's disease. Pretty, I like it. I like the colors. You know, it would be fun to do bacterial like plate art. Oh, I will hydrate. Thank you, thank you. I would like. Have you guys ever seen like people that make art on a petri dish? They like grab different bacteria that will look a different color and like make art on it, and it's so cool looking. I want to do something like that. I have been yapping for a bit, so I'm gonna hydrate even more right now. It feels really easy to just keep yapping because there's words on my screen and I'm just reading the words on my screen. So watch in the future. We will try to do some kind of like petri dish art. I am super inexperienced on this though, and so it's something I have to look up on how I can do. And also, I would have to figure out how to do this at home. I don't exactly have a lab, but you know, I would love to try something. We are on chapter 1.2 now. A systematic approach. And by the end of this section, you will be able to describe how microorganisms are classified and distinguished as unique species. Compare historical and current systems of taxonomy used to classify microorganisms. Making art and trying not to accidentally cause a disease outbreak. Once microbes become visible to humans with the help of microscopes, scientists began to realize their enormous diversity. Microorganisms vary in all sorts of ways, including their size, their appearance, and their rate of reproduction. To study this incredibly diverse new array of organisms, researchers needed a way to systematically organize them. Taxonomy. I really don't care about taxonomy, but we're going to read this so I can learn. Taxonomy is the classification, description, identification, and the naming of living organisms. Classification is the practice of organizing organisms into different groups based on their shared characteristics. The most famous early taxonomist was a Swedish botanist, zoologist, and physician. Triple threat, the botany, zoology, physician. Carolus Linnaeus. In 1735, Linnaeus published Systema Naturae, an 11-page booklet in which he proposed the Linnaean taxonomy, a system of categorizing and naming organisms using a standard format so scientists could discuss organisms using consistent terminology. 11 pages. He said, I need you guys to be standardized in 11 pages. It's kind of brilliant. He continued to revise and to add to the book, which grew into multiple volumes. There he is. What a lovely and elegant picture. The slight holding of the flowers. They look like the lily of the valley flowers with the little bells. I love his little hat. I love his collar. I love this tassel stuff. Beautiful. Absolutely beautiful. In his taxonomy, Linnaeus divided the natural world into 3 kingdoms: animal, plant, and mineral. Wait, you're telling me there was a mineral kingdom? There was a mineral kingdom? And we abandoned the mineral kingdom? That can't. We need to bring back calling microorganisms wee little beasties, and we also need to bring back the mineral kingdom. We diverge too far from historical science. Within the animal and plant kingdoms, he grouped organisms using a hierarchy of increasingly specific levels and sublevels based on their similarities. The names of the levels in Linnaeus' original taxonomy were kingdom, class, order, family, genus, and species. Species was and continues to be the most specific and basic taxonomic unit. I want to hear more about the mineral kingdom. Hi, thanks for checking in, Weetabix. Hello. Evolving trees of life. With advances in technology... Hello, Weetabix. Hi. With advances in technology, other scientists gradually made refinements to the Linnaean system and eventually created new systems for classifying organisms. In the 1800s, there was a growing interest in developing taxonomies that took into account the evolutionary relationships or phylogenies of all different species of organisms on Earth. You know, you're gonna think I'm not very educated because I keep mispronouncing words, but it's just because in my brain I read them more than I say them, and so I just forget. How they're pronounced. One way to depict these relationships is via a diagram called a phylogenetic tree, or tree of life. Oh! You know, that makes sense why it's called tree. It would be called tree of life. In these diagrams, groups of organisms are arranged by how closely related they are thought to be. In early phylogenetic trees, the relatedness of organisms was inferred by their visible similarities, such as the presence or absence of hair or the number of limbs. Now the analysis is more complicated. Yeah, I took a genetics course, like a animal biology genetics course, and I wish it was as simple as, yeah, does it have hair? Oh, does it have 2 legs? Yeah, they're related. It's so freaking complicated. I hated that class, and I did not stick around for it. Nope. I dropped that when I realized I don't need this. Don't need this. Now the analysis is more complicated. Today, phylogenetic analysis include genetic, biochemical, and embryological comparisons, as will be discussed later in this chapter. Linnaeus' tree of life contained just 2 main branches of all living things, the animal and plant kingdoms. Hey, what about the mineral kingdom? I feel like this book is throwing the mineral kingdom, away. I want to learn more about the mineral kingdom. We're gonna look up what the mineral kingdom is about after this. In 1866, Ernst Haeckel... My goodness, all these European names that I am butchering so hard. I'm sorry to my Europeans out there. I'm so sorry. A German biologist, philosopher, and physician. A biologist, philosopher, and physician. I don't think that's as cool as botanist, zoologist, physician. That's a good try. Proposed another kingdom, Protista, for unicellular organisms. He later proposed a 4th kingdom, Monera, for unicellular organisms whose cells lack nuclei. Like bacteria. That's me! I'm part of that kingdom. But I want to be in the mineral kingdom. I bet viruses were part of the mineral kingdom. We gotta bring back the mineral kingdom. Sure, it might not make sense because maybe he did put rocks in there. There'd be rocks and viruses in the mineral kingdom. But, you know, is that so wrong? Hi, Rivko, thanks for checking into the Petri dish. Nearly 100 years later, in 1969, American ecologist Robert Whitaker proposed adding another kingdom, fungi, in his tree of life. Oh man, the fungi get a tree, they get a kingdom, but the minerals can't. It's not fair. It's absolutely unright. Whitaker's tree also contained a level of categorization above the kingdom level. the empire or super kingdom level to distinguish between organisms that have membrane-bound nuclei in their cells, eukaryotes, and those that do not, prokaryotes. Empire prokaryota contain just the kingdom Monera. The empire eukaryota contain the other 4 kingdoms, fungi, protista, plantae, and animalia. Whittaker's 5-kingdom tree was considered the standard phylogeny for many years. Isn't it funny reading this? This almost sounds like a... It just sounds hilarious. They're just talking about kingdoms and empires, but it just feels... it doesn't feel like science. It just feels like some kind of, like, royal, like, fantasy. The empire prokaryota and their 4 kingdoms, fungi, protista, plantae, and animalia. Oh, viruses are not found in any of these trees. That is because they are not made up of cells, and thus it is difficult to determine where they would fit into a tree of life. The cowards, the absolute cowards. They bring back Mineral Kingdom. Bring it back. Carolus Linnaeus. Look how simple. Okay, they are just pretending that he didn't propose a Mineral Kingdom. They're like gaslighting me. They just talked about Mineral Kingdom and they're saying no, that's not a thing. How dare they? How dare they? Man, I feel like isn't this picture like they're a shrub next to the tree of life. Yeah, the viruses, they would be like right here, chillin'. Also, aren't these pictures just like a downgrade? Like, I feel like Carolus's picture looks so much cooler than Robert Whittaker's. Robert Whittaker could be any guy from like the 60s. I like the glorp emotes a lot. Yeah, it feels like the Plant Kingdom marched on the outer Animalia borders of Eukaryota after declaring war in 1830. It just feels really funny. I know, we need to like bring back getting ourselves painted and then saying, yes, this is my LinkedIn profile picture. It's me painted by some guy whose only job was to make art. And he gave me a funky little hat. Yeah, this could be my professor in undergrad. This could be like my dad. He just looks, he doesn't even look very science related. He could be anybody. Come on, Robert Whittaker. Maybe I should get like painted. I get a portrait. Looks like disappointed Stephen Colbert. Let's ignore the clinical focus. The role of genetics in modern taxonomy. Haeckel and Whittaker's trees presented hypotheses about the phylogeny of different organisms based on readily observable characteristics. But the advent of molecular genetics in the late 20th century revealed other ways to organize phylogenetic trees. Genetic methods allow for a standardized way to compare all living organisms without relying on observable characteristics that can often be subjective. Modern taxonomy relies heavily on comparing the nucleic acids, deoxyribonucleic acid, DNA, or ribonucleic acid, RNA, or proteins from different organisms. The more similar the nucleic acids and proteins are between two organisms, the more closely related they are considered to be. My god. Remember when it was just about, yeah, it's plant, yeah, it's animal, yeah, it's mineral? Now we're talking about the genetic, similarity. Genetics is so complicated. Bring back Mineral Kingdom. Yeah, now to test this can of worms here. In 1970s, American microbiologist Carl Woese discovered what appeared to be a living record of the evolution of organisms. He and his collaborator George Fox created a genetics-based tree of life based on similarities and differences they observed in the gene sequence coding for a small subunit ribosomal RNA, rRNA, or of different organisms. In the process, they discovered that a certain type of bacteria called Archaebacteria, now known simply as archaea, were significantly different from other bacteria and eukaryotes in terms of their small subunit rRNA gene sequences. rRNA. To accommodate this difference, they created a tree with 3 domains above level of kingdom: archaea, bacteria, and eukarya. Analysis of small subunit RNA gene sequences suggests archaea, bacteria, and eukaryotes all evolved from a common ancestral cell type. The tree is skewed to show a closer evolutionary relationship between archaea and eukarya than they have to bacteria. Interesting. We are here. This is actually... No, we're not here. Where am I? I haven't decided where I am yet. But I'm here. We're all here. Maybe we have to do like some genetic testing to figure out where we are in this tree. Who are our relatives? We have to find out who we're related to. Scientists continue to use analysis of RNA, DNA, and proteins to determine how organisms are related. One interesting and complicating discovery is that of horizontal gene transfer. Oh my gosh, I remember this term. When a gene of one species is absorbed into another organism's genome. I'm taking your genome. I'm just taking it. Horizontal gene transfer is especially common in microorganisms and can make it difficult to determine how organisms are evolutionarily related. Consequently, some scientists now think in terms of webs of life rather than trees of life. I almost read that as like weebs of life, and I was like, why are they talking about weebs? Not grandpas. We have to figure out what our where we sit on this. I have to do some thinking. I have to assess the bacteria buds and myself. Naming microbes. Ooh, that sounds fun. In developing his taxonomy, Linnaeus used a system of binomial nomenclature, a 2-word naming system for identifying organisms by genus and specific epithet. For example, modern humans are in the genus Homo, and have the specific epithet sapiens, and so their scientific name in binomial nomenclature is Homo sapiens. In binomial nomenclature, the genus part of the name is always capitalized, is followed by the specific epithet name, which is not capitalized. Both names are italicized. When referring to the species of humans, the binomial nomenclature would be Homo sapiens. I feel like they repeated themselves a lot there, but I get it. Taxonomic names in the 18th through 20th century were typically derived from Latin, since that was the common language used by scientists when taxonomic systems were first created. Today, newly discovered organisms can be given names derived from Latin, Greek, or English. Sometimes these names reflect some distinctive trait of the organism. In other cases, microorganisms are named after the scientists who discovered them. The archaeon, oh my gosh, Haloquatridum walsabi is an example of both of these naming schemes. The genus Haloquatridum describes the microorganism's saltwater habitat. Halo is derived from the Greek word for salt. Oh, as well as the arrangement of its square cells. Oh, quad. Salt quad. Ah. The species Walsby is named after... Oh, I see, it's just Walsby. But they added a random I there. Come on. They threw me off. It's named after Anthony Edward Walsby, the microbiologist who discovered Haloquatridum walsbii in 1980. While it might seem easier to give an organism a common descriptive name like red-headed woodpecker, if we can imagine with how that could become problematic, well, that would be too crazy, especially for things that just kind of do the same things. What happens when another species of woodpecker with red head coloring is discovered? The systematic nomenclature scientists use eliminates this potential problem by assigning each organism a single, unique 2-word name that is recognized by scientists all over the world. Yeah, like Nicoplasm nico, are recognized by scientists all over the world. In this text, we will simply abbreviate an organism's genus and species after its first mention. The abbreviated form is simply the first initial of the genus, followed by a period and the full name of the species. For example, the bacterium Escherichia coli is shortened to E. coli in its abbreviated form. You will encounter the same convention in other texts as well. The allowance for stuff named after people can be problematic, like the hedgehog gene. Oh yeah, that one's interesting. I know they got into a lot of trouble, right, with that specific gene? Or is it that one? Let me see. Is it? Oh right, yeah. There's like the Sonic the Hedgehog gene. It's literally called Sonic Hedgehog protein, or SHH. And it caused encephalopathy condition. Well, it was like the mutations of that gene, the hedgehog protein gene, were linked to a condition that causes like a type of encephalopathy, and which causes like brain defects, skull defects, facial defects. Yeah, you tell someone, I'm sorry, your brain is defective because you have a mutation in the Sonic the Hedgehog protein. Yeah, scientists and it was not very like well received that you would have to tell someone your Sonic the Hedgehog gene is mutated. It's I think I understand it. It's maybe it's not the place to have those kind of names. But I know they weren't obviously thinking that when they first named it. It wasn't until like it was actually linked to a disorder where you go, oh, I see. Maybe this is why we shouldn't have fun. Maybe this is why we shouldn't have fun here. Maybe we should just go back to naming things after ourselves, like Mr. Walsby here. Bergey's manuals. Whether in a tree or a web, microbes can be difficult to identify without easily observable macroscopic features like feathers, feet, or fur. Scientists must capture, grow, and devise ways to study their biochemical properties to differentiate and classify microbes. Despite these hurdles, a group of microbiologists created and updated a set of manuals for identifying and classifying microorganisms. First published in 1923 and since updated many times, Bergey's Manual of Determinative Bacteriology and Bergey's Manual of Systematic Bacteriology are the standard references for identifying and classifying different prokaryotes. Because so many bacteria look identical, methods used on non-visual characteristics must be used to identify them. For example, biochemical tests can be used to identify chemicals unique to certain species. Likewise, serological tests can be used to identify specific antibodies that will react against the proteins found in certain species. Ultimately, DNA and rRNA sequencing can be used for both identifying a particular bacterial species and for classifying newly discovered species. Makes sense. Makes sense. With same name, different strain. Oh, that's a good... This is also a good section to think about. How, like, there's many different strains just in the same species. Within one species of microorganisms, there can be several subtypes called strains. While different strains may be nearly identical genetically, they can have different attributes. The bacterium Escherichia coli is infamous for causing food poisoning and traveler's diarrhea. However, there are many different strains of E. coli, and they can vary in their ability to cause disease. One pathogenic E. coli strain that you may have heard of is E. coli 0157H7. In humans, infection from E. coli 0157H7 can cause abdominal cramps and diarrhea. Infection usually originates from contaminated food or water, particularly raw vegetables and undercooked meats. In the 1990s, there were several large outbreaks of E. coli 0157H7 thought to have originated in undercooked hamburgers. Not the hamburgers! The 1990s undercooked hamburgers epidemic. I did learn that, like, rare hamburgers, like, were much more common back in the day. I don't want to say back in the day because Back in the day sounds much longer, much long ago, like much longer than the 1990s, but it is back in the day. I feel like people are way more careful now about what temperature their hamburger is. I don't see a lot of people saying they want a rare burger unless it's somewhere they can really trust. And also, like, look at like fast food places now, like with burgers. Not that you're expecting much, but it's super well done, the patty. While E. coli's O157H7 and some other strains have given E. coli a bad name, most E. coli strains do not cause disease. In fact, some can be helpful. Different strains of E. coli found naturally in our guts help us digest our food, provide us with some needed chemicals, and fight against pathogenic microbes. Yeah, especially if you grind meat in your house. It's because ground beef is just so it's like sussy of a meat. You don't really know where, like, the surface, what part of the meat it came from, and like it could have touched things that it shouldn't have touched, and now it's all mixed in there. Versus like a cut of steak, which is like there's obviously the surface, and that could be contaminated, but the inside generally is not contaminated. Yeah, it's so much surface area open. Not good, not good. Oh, types of organisms. By the end of this section, you'll be able to list the various types of microorganisms and describe their defining characteristics, give examples of different types of cellular and viral microorganisms and infectious agents, describe the similarities and differences between archaea and bacteria, provide an overview of the field of microbiology. Okay, we're going to be able to do all of that, guys. Most microbes are unicellular and small enough that they require artificial magnification to be seen. However, there are some unicellular microbes that are visible to the naked eye, and some multicellular organisms that are microscopic. An object must measure about 100 micrometers to be visible without a microscope, but most microorganisms are many times smaller than that. For some perspective, consider that a typical animal cell measures roughly 10 micrometers across, but is still microscopic. Bacterial cells are typically 1 micrometer, and viruses can be 10 times smaller than bacteria. Oh. An atom. Now, this is a nanometer. Fullerene, lipids, protein, polio, flu, mitochondria, smallpox virus, bacteria, red blood cell, animal cell, plant cell, pollen, human egg, frog egg, scale, not to scale. Yeah. Wait. And then me. I'm on here too. I'm like, I'm far beyond their scale. They need a bigger scale to fit me in here. But I'm here too. I'm here. I'm not here. I'm like way over here. And I just remembered that giant unicellular organism. Have you guys seen it? It's like a giant, like, green sphere. And it's like, I think it's algae. It's just algae found in the ocean. And it could be like 4 centimeters, like, diameter. And it's just so funny looking. It's just like a giant green grape. And it's just a single-celled organism of algae. I love it so much. Yeah, it's so cool. I just want to, like, touch it. Whenever I see pictures of it, I just want to touch it so bad. And I want to eat it also because it looks like a grape, but I know it's probably it's do not eat the unicellular algae ball. Don't eat it. Don't eat it, guys. It's definitely not worth eating it. Units: meter, decimeter, centimeter, millimeter, micrometer, nanometer. My goodness. When you put like all the zeros, you can really feel like how tiny things are. You know, I would really struggle in my like general chemistry classes with conversions, like unit conversion. Something about my brain would never wrap around unit conversion. Oh, I bet organelles, there's no way they taste good, but I also am curious. Like, there's no way it tastes good, but also If I could try it, maybe I would want to try it. It's always like so disappointing how most things in the world are actually not that tasty. Like, barring food. It's almost like it's because those aren't things we're meant to eat. Most things are not meant for consumption, therefore, there's no reason for it to be tasty. But then it's sad. What do you mean the single celled algae ball is not supposed to be tasty? What a waste. What a waste of nature. Microorganisms differ from each other not only in size, but in structure, habitat, metabolism, and many other characteristics. While we typically think of microorganisms as being unicellular, there are also many multicellular organisms that are too small to be seen without a microscope. Some microbes, such as viruses, are even acellular. Yeah, aw. I like your emote, Caldonia. It's really cute. A decent chunk of early science was let me taste this and write it down. Yeah, I mean, can you blame them? They're probably like, okay, no one's ever tasted this before. Let me see if this is good. Let me see if this is worth it. One of these days, it's gonna be worth it, guys. We're gonna keep trying to taste these things until we find the things that are worth it. Microorganisms are found in each of 3 domains of life: archaea, bacteria, and eukarya. Microbes within the domains bacteria and archaea are all prokaryotes. Their cells lack a nucleus, whereas microbes in the domain of eukarya are eukaryotes. Their cells have a nucleus. Some microorganisms, such as viruses, do not fall within any of the 3 domains of life. In this section, we will briefly introduce each of the broad groups of microbes. Later chapters will go into greater depth about the diverse species within each group. Prokaryotic microorganisms. Bacteria are found in nearly every habitat on Earth, including within and on humans. Most bacteria are harmless or helpful. That's me. I'm the helpful and I'm harmless and helpful. Some are pathogens causing disease in humans and other animals. Bacteria are prokaryotic because their genetic material DNA is not housed within a true nucleus. Most bacteria have cell walls that contain peptidoglycan. Me, I'm peptidoglycan. I have no true nucleus. I'm just peptidoglycan. Bacteria are often described in terms of their general shape. Common shapes include spherical, coccus, rod-shaped, bacillus, or curved, spiralium, or vibrio. Figure 1.13 shows these examples. Ooh. The circle, the rod, the funky guy, the oval, the little swirly, little curly dude, and little curly dude again. I'm a big fan of the bacillus shape. I think those are kind of funny, but also the curly ones are cool too. This Coccobacillus one, I feel like it's not doing it much justice, but I get it. It's because they're all connected over here. Oh, hi. Hi, Aspirin. I'm glad you were able to stop by and say hi. It's okay if you can't stick around. I'll always be here in another day. I hope you have fun doing whatever you need to do. They have a wide range of metabolic capabilities and can grow in a variety of environments using different combinations of nutrients. Some bacteria are photosynthetic, such as oxygenic cyanobacteria and oxygenic green sulfur and green non-sulfur bacteria. These bacteria use energy derived from sunlight and fix carbon dioxide for growth. Other types of bacteria are non-photosynthetic, obtaining their energy from organic or inorganic compounds in their environment. Archaea are also unicellular prokaryotic organisms. Archaea and bacteria have different evolutionary histories as well as significant differences in genetics, metabolic pathways, and the composition of their cell walls and membranes. Unlike most bacteria, archaeal cell walls do not contain peptidoglycan, but their cell walls are often composed of a similar substance, called pseudopeptidoglycan. Like bacteria, archaea are also found in nearly every habitat on Earth, even extreme environments that are very cold, very hot, very basic, or very acidic. Some archaea live in the human body, but none have been shown to be human pathogens. Oh, is this the hot spring? Oh, yeah. I think that's so fascinating. There's just things living there. I mean, they're microorganisms. It's so pretty. Giant hole. Pretty colors. I like that. I like that a lot. That section. I liked learning about me, myself, the bacteria. And now we have to go to the eukaryotic microorganisms. Very lame. Very lame. The domain eukarya contains all eukaryotes, including unis or multicellular eukaryotes such as protists, fungi, plants, and animals. The major defining characteristic of eukaryotes is that their cells contain a nucleus. Boo, nucleus containers. Boo, boo. Algae. Oh, no. Protists are an informal grouping of eukaryotes that are not plants, animal, or fungi. Some algae are protists, and others are bacteria. All protozoa are examples of protists. Oh no. Oh no, the tentacles are getting me. Algae are mostly made up of protists that can be either unicellular or multicellular and vary widely in size, appearance, and habitat. Algal protists are surrounded by cell walls made up of cellulose, a type of carbohydrate. Algae are photosynthetic organisms that extract energy from the sun and release oxygen and carbohydrates into the environment. Cyanobacteria, a type of bacteria, is also considered an algae, but these organisms are bacterial prokaryotes and therefore have a peptidoglycan-based cell wall, unlike the cellulose-based cell wall of algal protists. Because other organisms can use the waste products of all algae for energy, algae are important in parts of many ecosystems. Many consumer products contain ingredients derived from algae, such as carrageenan or alginic acid, which are found in some brands of ice cream. Oh, interesting. Oh, I do know that. I do know that. It's like a stabilizer or like thing that thickens it, right? Salad dressing, beverages, lipstick, and toothpaste. A derivative of algae also plays a prominent role in the microbiology lab. Agar. Oh yeah, we love agar. A gel derived from algae can be mixed with various nutrients and used to grow microorganisms in a petri dish. Algae are also being developed as possible sources for biofuels. I had a conversation with a guy one time about likely pathogens and extremophile bacteria. He didn't take it well when I brought up that if he were hot enough for those to reproduce, he would literally be cooked. That's really funny. He'd be cooked. Not figuratively, he'd be literally cooked. Oh, pretty. They're so funny looking. I think it's the fact that it's a black background also makes it look even funkier. It's so fascinating. Protozoa are protists that make up the backbone of many food webs by providing nutrients for other organisms. Protozoa are very diverse. Some protozoa move with help from hair-like structures called cilia or whip-like structures called flagella. Yeah, flagella gang. Others extend part of their cell membrane and cytoplasm to propel themselves forward. These cytoplasmic extensions are called pseudopods, false feet. Some protozoa are photosynthetic, others feed on organic material. Some are free-living, whereas others are parasitic, only able to survive by extracting nutrients from a host organism. Most protozoa are harmless, but some are pathogens that can cause disease in animals or humans. Oh yeah, diatoms are so, I think they're so cool. Giardia lamblia. Lamblia? An intestinal protozoan parasite that infects humans and other mammals, causing severe diarrhea. Oh, it's so cool looking. I know it's like I'm anthropomorphizing it, but it looks like it's got like funny little legs and a tail and like a big body. So it's very funny. I'm looking at it completely wrong. It's just those are just the thing, like flagella and propelling it. It looks like it's got really nice legs. Well, too skinny, but like her funny little legs. Fungi. Fungi are also eukaryotes. Some multicellular fungi, such as mushrooms, resemble plants, but they are actually quite different. Fungi are not photosynthetic, and their cell walls are usually made of chitin rather than cellulose. Unicellular fungi, yeasts are included within the study of microbiology. Fungi. Yeah, it's a fun guy. There are more than 1,000 known species. Yeasts are found in many different environments from the deep sea to the human navel. the human navel. Some yeasts have beneficial uses, such as causing bread to rise and beverages to ferment, but yeast can also cause food to spoil. Some even cause diseases such as vaginal yeast infections and oral thrush. Some like crazy whiplash from like bread rise. Also vaginal yeast infections. Oh, yeast. Yeast is so crazy. We have an ad coming up. I'm gonna take this as a time to probably take a little mini stretch and also fill up my water again. So I'll be right back. Burp, burp, burp. I'm back with water. I think there's still like a minute left on the ad. I'm gonna just look at the funny pictures. Candida albicans is a unicellular fungus or yeast. This is the thing that causes yeast infections, right? Oh yeah, it is. Yeast infection, C. albicans. It's kind of funny looking. Blue. Tiny and blue. It does look like coccus bacteria. Like, it's all circular, too. This picture of the oranges right here, this is exactly what happens whenever I buy oranges. Like, a bag of oranges at my grocery store, I feel like always ends up looking just like this. Because at the bottom of the bag, there's just one bad orange, and I didn't see it. And now it's spread to all the other oranges. Okay, ad break over. Welcome back. Hi, we were just looking at the pictures. We were looking at the Candida albicans. It's funny looking because it looks just like coccus bacteria. but it's much bigger than Coccus. Anyway, let's continue. Other fungi of interest to microbiologists are multicellular organisms called molds. Molds are made up of long filaments that form visible colonies. Molds are found in many different environments, from soil to rotting foods to dank bathroom corners. Dank bathroom corners. I don't think it doesn't sound like a good thing to have a dank bathroom corner. The word dank, it's not meaning what it's meaning in my brain. Molds play a critical role in the decomposition of dead plants and animals. Some molds can cause allergies, and others produce disease-causing metabolites called myotoxins. Molds have been used to make pharmaceuticals, including penicillin, which is one of the most commonly prescribed antibiotics, and cyclosporin, used to prevent organ rejection following a transplant. If you are in my Discord, you'll see sometimes that I share stuff from the subreddit Moldly Interesting, because I can't help but find mold looks so fascinating. Have you ever seen, like, I've never let food go that bad to see some of the mold that these people are posting in Moldly Interesting. It's like growing hair. They have Tupperware containers growing hair, and I'm like, how did you forget your tuna salad in the back of your fridge for that long, where there's black hair growing all over it? I don't get it. But also, keep posting it. I want to keep seeing it. I can't look away. I'll show my friends, and they're going to be like, why are you showing me this? And I just think it's kind of neat. Helminths. Oh, ew, worms. It's morbidly fascinating. You can't look away. It's just also like, I don't get it. How do you do that? Okay, this is kind of gross looking. Don't look if you don't want to see worms. Multicellular parasitic worms are called helminths are not technically microorganisms, as most are large enough to see without a microscope. However, these worms fall within the field of microbiology because diseases caused by helminths involve microscopic eggs and larvae. One example of a helminth is a guinea worm, or Dracunculus medinensis, which causes dizziness, vomiting, diarrhea, and painful ulcers on the legs and feet when the worm works its way out of the skin. Ew! Ugh. Kind of fascinating, though. Infection typically occurs after a person drinks water containing water fleas infected by the guinea worm larva. In the mid-1980s, there were an estimated 3.5 million cases of guinea worm disease, but the diseases have been largely eradicated. In 2014, there were only 126 cases reported, thanks to the coordinated efforts of the World Health Organization and other groups committed to improvements in drinking water sanitation. Damn. Like, in current year, it makes me sad to see things like that. Just because, like, yeah, these organizations and efforts, they're so important. Look, they eradicate diseases. It's almost like we should really be funding these things. I worked for a pharmacy one time. There was a bottle of amoxicillin that got reconstituted and somehow rolled behind a counter. We found it after long enough there was something growing the antibiotics. Wow. Wait, that's insane. I would not have thought that something could be growing in it, but I guess it's got water. I feel like when you add water to anything, life will find a way. Life will find a way to grow. The beef tapeworm. Hania saginata infects both cattle and worms. My goodness, 4 to 10 meters. It's insane how long this is. 4 to 10 meters. All in the digestive system. A guinea worm. Oh, it's so tiny and gross. Ugh. You can wind it on a matchstick. Ugh. Yuck. Viruses are acellular microorganisms, which means they are not composed of cells. Essentially, a virus consists of proteins and genetic material, either DNA or RNA, but never both, that are inert outside of a host organism. However, by incorporating themselves into a host cell, viruses are able to co-opt the host's cellular mechanisms to multiply and infect other hosts. Viruses can infect all types of cells, from human cells to the cells of other microorganisms. In humans, viruses are responsible for numerous diseases, from the common cold to deadly Ebola. However, many viruses do not cause disease. I actually didn't know that. I wouldn't have thought many don't. But I also guess it makes sense. The coronavirus family can cause respiratory infections like the common cold, severe acute respiratory syndrome, and Middle East respiratory syndrome. Here they are viewed under a transmission electron microscope. Interesting. It's got the little things. I feel like after COVID, it's very this is probably one of the most distinct like viral silhouettes. Like even people unaware of any scientific knowledge can tell you it's COVID. Like the little spiky guys, spiky guys, that's COVID. Yeah, I guess that's true. Most common virus is probably, it's like a bacteriophage that doesn't do, like, it's not causing a disease for us. Okay, and this is Ebola, member of the filovirus family, as visualized using a TEM. Interesting. Yeah, I guess this does not have anywhere near the iconic shape of, like, coronavirus family. Microbiology as a field of study. Microbiology is a broad term that encompasses the study of all different types of microorganisms. But in practice, microbiologists tend to specialize in one of the several subfields. For example, bacteriology is the study of bacteria, mycology is the study of fungi, protozoology is the study of protozoa, parasitology is the study of helminths and other parasites, and virology is the study of viruses. Immunology, the study of the immune system, is often included in the study of microbiology because host pathogen interactions are central to our understanding of infectious disease processes. Microbiologists can also specialize in certain areas of microbiology, such as clinical microbiology, environmental microbiology, applied microbiology, or food microbiology. Ooh, food microbiology. I used to think when I would, was trying to decide what kind of field of science I wanted to go into, I thought food science was a very interesting topic and that maybe I wanted to study that. But I didn't. The schools I applied to at that time, they didn't have anything that felt like I could study that, so I never did. But I followed some girl on YouTube. She did a food science program at Cornell. And when I saw what she did, and then also what she does now as a job, I realized that's probably not something I would have enjoyed. Apparently, it's a lot of business marketing that you end up going to. I'm sure there's much more science related applications, but her job sounds a lot like how it's like an interface of okay, we have science safety, and then how do we make sure we can maximize profits? And I feel like you'd probably run into that kind of job more often than not, especially in our current landscape. Just people wanting to maximize profits with the least amount of food safety. Yeah, I'm sure there's food science labs out there. There has to be, because someone has to figure out how these things are food safe, how things are working and how they're developing. Also, it seems like I might have avoided a field where it seems like things aren't going so well, at least in America. At least in terms of if you wanted to study food science regulation. Let's see. A virologist samples eggs from this nest to be tested for influenza A virus, which causes avian flu. Yeah, I'm sure. I guess pharmaceuticals probably have this. I think this is a thing in a lot of scientific fields. I know a lot of people that were hardcore chemists. They study a lot about chemistry, and then they went on to go to do a sales rep job. And if they do really well in it because they can talk very in-depth and knowledgeable about specific aspects of the machine that they're trying to sell. It doesn't sound very fun to me. Or interesting, but you know, someone has to do it. Who gets commission versus who gets a bonus based on regional performance, and who could make what sort of claims, like FDA-approved indications versus off-brand use. Oh, interesting. Ah, that's very interesting. Hi, Vixie! Hi. I'm glad you're able to make it to the stream. Eye on ethics. Ooh, ethics section, guys. Ethics section. In the 1940s, the US government was looking for a solution to a medical problem: the prevalence of sexually transmitted diseases among soldiers. Several now infamous government-funded studies used human subjects to research common STDs and treatments. In one, oh gosh, I know exactly what this this section is talking about. It's not a very, it's such a sad section. In one such study, American researchers intentionally exposed more than 1,300 human subjects in Guatemala to syphilis, gonorrhea, and chancroid to determine the ability of penicillin and other antibiotics to combat these diseases. Subjects of the studies included Guatemalan soldiers, prisoners, prostitutes, and psychiatric patients, none of whom were informed that they were taking part in the study. Researchers exposed subjects to STDs by various methods, from facilitating intercourse with infected prostitutes to inoculating subjects with the bacteria known to cause the diseases. My gosh, it's so terrible. It's absolutely absolutely messed up. Yeah, they could have brought up Tuskegee. This latter method involved making a small wound on the subject's genitals or elsewhere on the body and then putting bacteria directly into the wound. In 2011, a U.S. government commission tasked with investigating the experiment revealed that only some of the subjects were treated with penicillin, and 83 subjects died by 1953, likely as a result of the study. So sad. Unfortunately, this is one of many horrific examples of microbiology experiments that have violated basic ethical standards. Even if this study had led to a life-saving medical breakthrough, it did not. Parentheses, it did not. Good parentheses. Few would argue that its methods were ethically sound or morally justifiable, but not every case is so clear-cut. Professionals working in clinical settings are frequently confronted with ethical dilemmas, such as working with patients who decline a vaccine or a life-saving blood transfusion. These are just two examples of life and death decisions that may intersect with the religious and philosophical beliefs of both the patient and healthcare professional. No matter how noble the goal, microbiology studies and clinical practice must be guided by a certain set of ethical principles. Studies must be done with integrity. Patients and research subjects provide informed consent, not only agreeing to be treated or studied, but demonstrating an understanding of the purpose of the study and any risk involved. Patients' rights must be respected. Procedures must be approved by the Institutional Review Board. When working with patients, accurate record keeping, honest communication, and confidentiality are paramount. Animals used for research must be treated humanely, and all protocols must be approved by an institutional animal care and use committee. These are just a few of the ethical principles explored in the Eye of Ethics boxes throughout this book. Oh man. Whoever wrote this section, you can absolutely tell how passionate they are about ethics. They listed every single thing. They were like, yeah, you need to make sure you're in an institutional review board. Everyone must be informed. Everyone must be knowledgeable about what they're doing. They need to be put in if you're using animals, they have to use the IACUC. Good on whoever wrote this section. I really liked it. The religious objections to blood products. Oh yeah, I know it's really hard. Like, nothing about any of this is easy. It's rough. I think that it's so sad. This experiment they're talking about, and other experiments that have happened in science, it did so much harm, and still does do a lot of harm to this day on people's ability to trust science. There are a lot of people in America, especially minorities, who feel like they can't trust science because they were used unknowingly many times in history, like unfairly by science. They were just test subjects. It's not right. Really. Yeah, though, like, when someone tells me they don't trust science and they bring up experiments where they feel like science abused uh the level of professionality they have and the trust they feel like they feel like they can't trust science anymore because of that, I understand. And it's really hard to discuss after that. I just have to kind of tell them, you know, things are going a lot better. There are a lot more like safe, like, protocols in place to prevent what happened in the past. But it's sometimes still just not enough. And it's because this is still really recent. When you think about how 83 subjects died by 1953, there are people still alive who were affected by this. They might have not been like directly affected, but they might have known someone, like they might be related to someone who was affected by this. So to tell them they can trust science after they saw what happened, maybe firsthand, it's really like, I don't know how to do that. It was really rough, especially during the pandemic. I wanted to tell people you can believe in what they're telling you about the pandemic, but there was a lot of people still like apprehensive. Okay, clinical focus. Oh wait, they're bringing back the case from earlier. Earlier there was a case with meningitis, and it didn't look like there was any bacteria in the meningitis like sample or in the CSF sample. So now we're reading part 2 of what happened next to Cora. Cora's CSF sample shows no signs of inflammation or infection, as would be expected with a viral infection. However, there's a high concentration of a particular protein, 1433 protein, in her CSF. An EEG of her brain is also abnormal. The EEG resembles that of a patient with a neurodegenerative disease like Alzheimer's or Huntington's. But Cora's rapid cognitive decline is not consistent with either of these. Instead, her doctor concludes that Cora has Creutzfeldt-Jakob disease. Oh, that sucks. That sucks. A type of transmissible spongiform encephalopathy. That's not good. CJD is an extremely rare disease with only about 300 cases in the US each year. It is not caused by a bacterium, fungus, or virus, but rather by prions. Yes, spongiform encephalopathy is it's a great term. You guys know about prions? Prions are like they're so insane. Which prions mentioned. Guys, prions. They do not fit into any particular category of microbe. Like viruses, prions are not found on the tree of life because they are acellular. Prions are extremely small, about 1/10 the size of a typical virus. Whoa, I didn't realize they're that small, but I guess that would make sense. They contain no genetic material and are composed solely of a type of abnormal protein. They are really scary, but it's also like they're so fascinating. Creutzfeldt-Jakob disease can have several different causes. It can be acquired through exposure to the brain or nervous system tissue of an infected person or animal. Consuming meat from an infected animal is one way such exposure can occur. There have also been cases of exposure to CJD through contact with contaminated surgical equipment, and from cornea and growth hormone donors who unknowingly had CJD. Wow, I didn't know that. That's insane. Can you easily decontaminate your surgical equipment with it? I guess they're a type of protein, so likely. In rare cases, the disease results from a specific genetic mutation that can sometimes be hereditary. However, in approximately 85% of patients with CJD, I'd rather just say Creutzfeldt-Jakob disease. The cause of the disease is spontaneous or sporadic and has no identifiable cause. Yeah, I would think it probably doesn't autoclave off because it's a it's the type of protein it is pretty unique. I guess it would make sense how like there could possibly be contamination, but that means that's still kind of insane. I'm guess it's obviously not that common. You don't really hear about that. But what an unlucky like deal of hands if that's how you got it. Based on her symptoms and their rapid progression, Cora is diagnosed with a sporadic CJD. Don't. That's just Twitch for you. It'll just switch everything to an emoji. Don't worry about it. I can still read it fine. It looks funnier than it should be. Similar to kind of like the Sonic the Hedgehog thing. It might be like inappropriate in some discussion. Even if you can disinfect like crazy, they can still be transmitted. Insane. Yeah, it's insane that it can be transmitted. Unfortunately for Cora, CJD is a fatal disease for which there is no approved treatment. Approximately 90% of patients die within 1 year of diagnosis. Her doctors focus on limiting her pain and cognitive symptoms as her disease progresses. 8 months later, Cora dies. Her CJD diagnosis is confirmed with a brain autopsy. Oh, that's so sad. In the first box, we read that she just was having headaches, a stiff neck, and she was having a bit of like confusion. It's insane that it like progressed this far into that it was sporadic CJD. Poor Cora. I don't want to do all these questions. I don't want to critically think. But maybe we already finished one chapter. I didn't know we were gonna finish a chapter, but I kind of want to keep reading. I'm still in the mood to keep reading. How we see the invisible world. Maybe I'll post the quiz questions like on Twitter or my Discord, and I can grade you. Let's see. How we see it. Different types of microscopy are used to visualize different structures. Bright field microscopy left renders a darker image on a lighter background, producing a clear image of these bacillus anthracis cells and cerebrospinal fluid. The rod-shaped bacterial cells are surrounded by larger white blood cells. So you see these are like the little bacillus cells, and those are the white blood cells. Dark field microscopy right increases contrast, rendering a brighter deal of a darker background, as demonstrated by this image on the bacterium Borrelia burgdorferi, which causes Lyme disease. Look at it, it's all spindly. When we look at a rainbow, its colors span the full spectrum of light that the human eye can detect and differentiate. Each hue represents a different frequency of visible light, processed by our eyes and brains and rendered as red, orange, yellow, green, or one of the many other familiar colors that have always been part of the human experience. But only recently have humans developed an understanding of the properties of light that allow us to see images in color. Over the past centuries, we have learned to manipulate light to peer into previously invisible worlds. Too small or too far away to be seen by the naked eye. Through a microscope, we can examine microbial cells using various techniques to manipulate color, size, and contrast in ways that can help us identify species and diagnose disease. Figure 2.1 illustrates how we can apply the properties of light to visualize and magnify images. But these stunning micrographs are just two examples of the numerous types of images we are now able to produce with different microscopic technologies. This chapter explores how various types of microscopes manipulate light in order to provide a window into the world of microorganisms. By understanding how various kinds of microscopes work, we can produce highly detailed images of microbes that can be useful for both research and clinical applications. Yeah, if you don't like using those, you can always turn off 7TV. I think it just automatically puts it if you use the word that the emote is based on. You'll miss some emotes, but it doesn't, I don't think it's the end of the world. So let's look at the properties of light. Clinical focus. Oh no, is it gonna be another sad story? Part 1. Cindy, a 17-year-old counselor. Oh no, she's 17. Please don't do any. I hope nothing bad happens to you, Cindy. Cindy, a 17-year-old counselor at a summer sports camp, scraped her knee playing basketball 2 weeks ago. At the time, she thought it was only a minor abrasion that would heal, like many others before. Instead, the wound began to look like an insect bite and has continued to become increasingly painful and swollen. No! No, Cindy! The camp nurse examines the lesion and observes a large amount of pus oozing from the surface. Concerned that Cindy may have developed a potentially aggressive infection, she swabs the wound to collect a sample from the infection site. Then she cleans out the pus and dresses the wound, instructing Cindy to keep the area clean and to come back the next day. When Cindy leaves, the nurse sends the sample to the closest medical lab to be analyzed under a microscope. Oh my gosh, we're on the edge of our seats waiting to see what happens to Cindy. God, I hope her leg isn't like amputated or something in part 2. Visible light consists of electromagnetic waves that behave like other waves. Hence, many of the properties of light that are relevant to microscopy can be understood in terms of light's behavior as a wave. An important property of light waves is the wavelength, or the distance between one peak of a wave and the next peak. The height of each peak, or depth of each trough, is called the amplitude. In contrast, the frequency of the wave is the rate of vibration of the wave, or the number of wavelengths within a specified time period. Yeah, the camp nurse going, I need to send this to the closest medical lab stat is crazy. I feel like in most situations, would the camp nurse not just go, ooh, that looks bad, let's put a band-aid on it, and maybe I'll give you some Tylenol and come back tomorrow if you need a new band-aid. Good on this camp nurse. Also, this section is so physicsy, but we're gonna get through this. We will endure this for the future microbiology clinical sections. We must get through the tough times to get to the fun times. I'm feeling polymicrobial abscess coming on. Yeah. Interactions of light. Light waves interact with materials being reflected, absorbed, or transmitted. Reflections occur when a wave bounces off a material. For example, a red piece of cloth may reflect red light to our eyes while absorbing other colors of light. Absorbance occurs when a material captures the energy of a light wave. In the case of a glow-in-the-dark plastics, the energy from light can be absorbed and later re-emitted as another form of phosphorescence. I didn't actually know that's how that's how glow-in-the-dark stuff works. Okay, that's kind of cool. Transmission occurs when a wave travels through a material like light through glass. The process of transmission is called transmittance. When a material allows a large portion of light to be transmitted, it may do so because it is thinner or more transparent, having more transparency and less opacity. I feel like that concept is confused so often when people mistakenly call something like they're saying they say like increase opacity when they don't mean that at all. Especially in games. Figure 2.3 illustrates the difference between transparency and opacity. Yes, this is transparent. This is opaque. Light's not transmitted through this, light's transmitted through that. Thank you for the pictures, Wafer X of Flickr. Light waves can also interact with each other by interference, creating complex patterns of motion. Dropping 2 pebbles into a puddle causes the waves on the puddle's surface to interact, creating complex interference patterns. Light waves can interact in the same way. When I learned about that, topic of you drop pebbles and then they cancel their waves out, I was like, whoa, crazy. In addition to interfering with each other, light waves can also interact with small objects or openings by bending or scattering. Oh, my computer was not happy right there. This is called diffraction. Diffraction is larger when the object is smaller relative to the wavelength of the light. The distance between 2 consecutive peaks of a light wave, often when waves diffract in different directions around an obstacle or opening, they will interfere with each other. Man, that was a lot of physics. And it's still going, but We will get through this. I'm learning many, many things. In the context of microscopy, refraction is perhaps the most important behavior exhibited by light waves. Refraction occurs when light waves change direction as they enter a new medium. Different transparent materials transmit light at a different speed. Thus, light can change speed when passing from one material to another. This change in speed usually also involves a change in direction, refraction, with the degree of change dependent on the angle of incoming light. The white light goes zoom, and then goes boom, and now it's up there. Opaque, transparent, refracted. This concept is actually pretty important when you're drawing, you're coloring. You should really know where your light source is coming from and how it might be reflected in any way. But it's complicated. It's complicated to express that when you're drawing. Let me take a sip of water. The extent to which a material slows transmission speed relative to empty space is called the refractive index of the material. Large differences between the refractive indices of two materials will result in a large amount of refraction when light passes from one material to the other. For example, light moves much more slowly through water than through air, so light entering water from air can change direction greatly. We say that the water has a higher refractive index than air. This straight pole appears to bend at an angle as it enters water. This optical illusion is due to the large difference between the refractive indices of air and water. You see, does a little funky trick where it's ooh, it looks like it's bending, ooh, but it's actually not, ooh, ooh. When light crosses a boundary into a material with a higher refractive index, its direction turns to be closer to perpendicular to the boundary, i.e. more toward a normal to that boundary. This is the principle behind lenses. We can think of a lens as an object with a curved boundary, or a collection of prisms that collects all of the light that strike it and refracts it so that it meets at a single point called the image point, the focus. A convex lens can be used to magnify because it can focus at closer range than the human eye, producing a larger image. Concave lenses and mirrors can be used in microscopes to redirect the light path. Figure 2.6 shows the focal point, the image point where the light entering the lens is parallel, and the focal length, the distance to the focal point for convex and concave lenses. My god. I see, I understand, I am understanding. This is kind of like what you use for glasses, I guess. The human eye contains a lens that enables us to see images. This lens focuses the light reflecting off of objects in front of the eye onto the surface of the retina, which is like a screen in the back of the eye. Artificial lenses placed in front of the eye, contact lenses, glasses, or microscopic lenses, focus light before it is focused again by the lens of the eye, manipulating the image that ends up on the retina, e.g. by making it appear larger. Images are commonly manipulated by controlling the distances between the object, the lens, and the screen, as well as the curvature of the lens. For example, for a given amount of curvature, when an object is closer to the lens, the focal points are farther from the lens. As a result, it's often necessary to manipulate these distances to create a focused image on a screen. Similarly, more curvature creates image points closer to the lens and a larger image when the image is in focus. This property is more often described in terms of focal distance or distance to the focal point. I see. Naruhodo, naruhodo. They yapped so much about lenses. It's kind of crazy, but I get it. I mean, you're using lenses to look at these things, so they should probably explain how they work. I can't say I was ever curious about why I can see the things in the microscope, but that's just me. But it's good to learn. I understand now. I completely get it. Electromagnetic spectrum and color. Visible light is just one form of electromagnetic radiation, a type of energy that is all around us. Other forms of EMR include microwaves, X-rays, and radio waves, among others. The different types of EMR fall on the electromagnetic spectrum, which is defined in terms of wavelength and frequency. The spectrum of visible light occupies a relatively small range of frequencies in between infrared and ultraviolet. The classic. If you've been in a general, not a general chemistry, But a physical chemistry class, this is just like pasted like everywhere because they really wanted us to know this. It was really burned into my brain. This specific like where the frequencies are and what these are. Yeah. I can't say I find this stuff interesting. I've talked about in like my science education background, and math and physics related aspects have always been like my least favorite part of science. Someone could say the microscopes are programmed to show stuff because bacteria are a fiction that big pharma uses to sell antibiotics. You're exactly right. I have seen that argument before. I have seen people clearly not understand what a microscope is and say it's fake or that we can't trust what we see in a microscope. And it blows my mind. It's definitely up there with the people that go I talked about this example before, but the people that put like a blanket over their mirror and then they'll like wave their hand and then show that the mirror is like shows them waving their hand behind the mirror, and they're like, how does the mirror know that I'm waving my hand? And they're like trying to say like the mirror is like watching you from all over the place. And like the mirror isn't the mirror is not watching that you. The mirror is doing nothing. The mirror is just a thing there. It's not looking at you. But yes, we have to be aware of those people, like you said. There has to be education on how things work, or else you have to start at step 0 sometimes before you can help get them to the point understanding why what they're saying doesn't make sense. A cheap red laser would show them how the mirror is seeing them. I just think the aspect of people going, how does the mirror see me, is so funny. It's way funnier than it like should be. Something about like saying that the mirror saying sentences that make it seem like the mirror is looking at you is just so hilarious. It's just like being funny. But no, there's people that go, the mirror is looking at me. It's seeing me. Whereas wavelengths represent the distance between adjacent peaks of a light wave, frequency in a simplified definition represents the rate of oscillation. Waves with higher frequencies have shorter wavelengths and therefore have more oscillations per unit time than lower frequency waves. Higher frequency waves also contain more energy than lower frequency waves. This energy is delivered as elementary particles called photons. Woo! Give it up for the photons! Higher frequency waves deliver more energetic photons than lower frequency waves. Woo! Woo! Higher frequency waves! Woo! Photons with different energies interact differently with the retina. In the spectrum of visible light, each color corresponds to a particular frequency and wavelength. The lowest frequency of visible light appears as the color red, whereas the highest appears as the color violet. When the retina receives visible light of many different frequencies, we perceive this as white light. However, white light can be separated into its component colors using refraction. If we pass white light through a prism, different colors will be refracted in different directions, creating a rainbow-like spectrum on a screen behind the prism. This separation of colors is called dispersion, and it occurs for a given material. The refractive index is different for different frequencies of light. Pretty prism make pretty colors. Ooh! Certain materials can refract non-visible forms of EMR and in fact transform them into visible light. Certain fluorescent dyes, for instance, absorb ultraviolet or blue light and then use the energy to emit photons of a different color, giving off light rather than simply vibrating. No more vibration. Brrr. This occurs because the energy absorption causes electrons to jump to higher energy states, after which they then almost immediately fall back down to their ground states, emitting specific amounts of energy as photons. Not all of the energy is emitted in a given photon, so the emitted photons will be of the lower energy and thus of lower frequency than the absorbed ones. Thus, a dye such as Texas red may be excited by blue light and emit red light, or a dye such as fluorescent Fitzy. I'm not going to say that name. I'm not going to say what Fitzy's supposed to be. May absorb invisible high-energy ultraviolet light and emit green light. In some materials, the photons may be emitted following a delay after absorption. In this case, the process is called phosphophorescence. Glow-in-the-dark plastic works by using phosphophorescent material. My gosh, when they talked about Texas red and Fitzy, I just had like my brain went boom. Flow cytometry. I used to, yeah, fluorescence. It's crazy how many different dyes there are. One of the, like, hard parts of my, like, PhD thesis before I left was making panels that worked to find all these different biomarkers for, flow cytometry. And it would be so complicated. It was just like a puzzle to go, okay, I need to find something here, and then, like, but it can't interact with this dye, so I have to make sure it's not that close. However, that overlaps with this dye, so I can't use it, and it was really complicated, but also really rewarding when you were able to figure out how to make it work. Also, I realized, like, when I started doing that, the flow cytometry core of, I'm gonna assume, like, most schools, you could just go to them and they'll figure it out for you for, like, a charge. Glows like, wow, that's toxic. I love that kind of color, the toxic yellow green. It's like comically like, ooh, that's not good for you. That's really bad for you. The fluorescent dyes absorbed by the bovine pulmonary artery endothelial emit brilliant colors when excited by UV light under a fluorescence microscope. Once made a cosplay beaker of chemicals was just oh, I bet that would look really cool though. Like, I feel like that's exactly what, like, science looks like. Toxic yellow-green with smoke and bubbles. That's what people would say science looks like. The nuclei are stained blue with, you're not also gonna make me say what DAPI is. I'm just gonna say DAPI. There's the nuclei are stained with DAPI. Microtubules are marked by an antibody bound to Fitzy, and actin filaments are labeled red with, I don't know, Tritzy, but I'm gonna guess Tritzy. DAPI, Fitzy, Tritzy. I like the word. I like these names of these dyes. DAPI is just such a cute name. DAPI, 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 Fitzy. They just sound fake. Magnification, resolution, and contrast. Microscopes magnify images and use the properties of light to create useful images of small objects. Magnification is defined as the ability of a lens to enlarge the image of an object when compared to the real object. For example, a magnification of 10x means that the image appears 10 times the size of the object as viewed with the naked eye. Greater magnification typically improves our ability to see details of small objects, but magnification alone is not sufficient to make the image more useful. It is often useful to enhance the resolution of objects. The ability to tell that two separate points or objects are separate. A low-resolution image appears fuzzy, whereas a high-resolution image appears sharp. This seems like really straightforward, but I do remember learning this in bio class, and I was sitting next to a friend, and she didn't really understand this. So it's like, oh, okay, I get it. You really need to break down all these aspects, because you got to make sure people understand what they're learning, what they're doing. Two factors affect resolution. The first is wavelength. Shorter wavelengths are able to resolve smaller objects. Thus, an electron microscope has a much higher resolution than a light microscope, since it uses an electron beam with a very short wavelength, as opposed to the long wavelength visible light used by a light microscope. The second factor that affects resolution is numerical aperture, which is a measure of a lens's ability to gather light. The higher the numerical aperture, the better the resolution. Even when a microscope has high resolution, it can be difficult to distinguish small structures in many specimens because microorganisms are relatively transparent. The third is how much you paid for your graphics card. Yes, exactly. I have a setup to use microscope objectives in my digital camera. Fun to take pictures. Oh, that's really cool. I've seen like pictures of that online. Like when they take like nice digital photos of things, like really like zoomed in. I think it's like so fascinating. It looks so nice. Even when a microscope has high resolution, it can be difficult to distinguish small structures in many specimens because microorganisms are relatively transparent. It's often necessary to increase contrast to detect different structures in a specimen. Various types of microscopes use different features of light or electrons to increase contrast. Visible differences between the parts of a specimen. Additionally, dyes that bind to some structures but not others can be used to improve the contrast between images of relatively transparent objects. Oh yeah, I'd love to hear what's a cool thing you were able to photograph. What are some fun things you got? Thank you for the hydration. We've been yapping a lot. And it's just so fun. Something about reading this textbook to you guys is just so fun. Yap 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 yap. Rotifers and a tardigrade. Oh, that's cool. That's really fun. A small mineral growing from Lincoln's head on the back of a penny. Oh, that sounds so cool. That sounds really cool. That's insane. I bet you really couldn't tell that he had a little mineral. God, I bet that mineral was. We gotta bring back Mineral Kingdom. Sorry to bring it back up again, but we're missing out with no Mineral Kingdom. Peering into the invisible world. Some of the fundamental characteristics and functions of microscopes can be understood in the context of history of their use. Oh, we're going back to history lessons. Italian scholar Girolamo Fracastoro. That's such an Italian-sounding name. I'm not Italian, but just the letters put together. Is regarded as the first person to formally postulate, not state, he postulated that disease was spread by tiny invisible seminaria, or seeds of the contagion. Ooh, I like that word. The seeds of contagion. In his book De Contagion, he proposed that these seeds could attach themselves to certain object, which he called foams. Foams that supported their transfer from person to person. However, since the technology for seeing such tiny objects did not yet exist, the existence of seminaria remained hypothetical for a little over a century, an invisible world waiting to be revealed. Oh man. He was right this whole time. A century goes by and he doesn't even get to see that he was right. Early microscope. It's our boy Antony van Leeuwenhoek, sometimes hailed as the father of microbiology, is typically credited as the first person to have created microscopes powerful enough to view microbes. Born in the city of Delft in the Dutch Republic, Van Leeuw began his career selling fabrics. However, he later became interested in lens making, perhaps to look at threads. I like how they put a parentheses to go like, we really don't know why he went from fabric selling to looking at lenses. Maybe to look at threads. We don't know. And his innovative techniques produced microscopes that allowed him to observe microorganisms as no one had before. In 1674, he described his observations of single-celled organisms whose existence was previously unknown in a series of letters to the Royal Society of London. His report was initially met with skepticism, but his claims were soon verified and he became something of a celebrity in the scientific community. He's something of a celebrity. What a sick picture of him. What's this? Oh right, this was the guy, the wee little beasties. I love this guy. I'm so sad that I'm so sad. He was onto something by calling them wee little beasties. Yeah, it was early. Anyone could just do a hobby. He's just wasn't that like isn't this time period just so nice? You could dedicate your whole life to looking at fabric and then all of a sudden pivot to lens making. Just cuz. Just cuz. Animalcules. Animalcules are also a really good name. I think I'm more partial to wee little beasties. If you want to refer to me as a wee little beastie, I won't be mad. Even though his microscopes were simple microscopes, as seen in this replica. Whoa, is this all he had? I'm trying to understand what I'm even looking at. I guess it's just... Oh, I see. That's the lens. Oh. He's kind of a genius if you think about it. Yeah, it's just like pinhole optics. It's very smart. I'm surprised I'll be honest, I'm surprised that it was able to like see a single-celled organism. Oh, that's more sushi. Yes, I'm maybe a little bit more sushi than bacteria right now. It'll clear through though. I'll be back to being mostly bacteria. Galileo Galilei, also the pioneers of microscopy. Well, I think this picture is pretty good. While Van Leeuw is credited with the discovery of microorganisms, others before him had contributed to the development of the microscope. These included eyeglass makers in the Netherlands in the late 1500s, as well as the Italian astronomer Galileo, who used a compound microscope to examine insect parts. Ah. Yeah. I drank water and then it went, like, it did not enjoy being drunken. Whereas Van Leeuw used a simple microscope in which light is passed through just one lens, Galileo's compound microscope was more sophisticated, passing light through two sets of lenses. Two, not one, two! His contemporary, Van Leeuw's contemporary, the Englishman Robert Hooke, also made important contributions to microscopy. Oh, I know about this guy. That was like a whole section in one of my courses about his microscopes. Publishing in his book Micrographia many observations using compound microscopes, viewing a thin sample of cork through his microscope, he was the first to observe the structures that we now know as cells. Hooke described these structures as resembling honeycomb and as small boxes or bladders of air. Bladders of air is a funny, like, word. Noting that each cavern, bubble, or cell is distinct from others. In Latin, cell literally means small room. Ah. They likely appeared to Hooke to be filled with air because the cork cells were dead, with only the rigid cell walls providing the structure. His little microscope. I remember seeing this exact picture. Both of these engravings are from his seminal work Micrographia. What a beautiful picture. You know, it was also crazy back then. I feel like a lot of scientists were just amazing artists. And nowadays, I think that that's not really the case. They're not sketching their own graphics now. Yeah, kerosene burning microscope. Pretty wild. That's like so funny looking. I feel like if you saw this today, you would not think of this as the microscope. Yeah, I don't think it's kerosene. I don't know. I can't tell what it is. I see. This is like what he was looking at. The cork cells. Who invented the microscope? I bet this is very contentious. While Antony van Leeuwenhoek and Robert Hooke generally received most of the credit for early advances in microscopy, neither can claim to be the inventor of the microscope. Some argue that this designation should belong to Hans and Zacharias Janssen, Dutch spectacle makers who may have invented the telescope, the simple microscope, and the compound microscope during the late 1500s or early 1600s. Gas. Unfortunately, little is known for sure about the Jansons, not even the exact dates of their births and deaths. Oh, sad, sad, so unknown. Unfortunate. The Jansons were secretive about their work and never published. It is also possible that Jansons did not invent anything at all. Their neighbor Hans Lippershey also developed microscopes and telescopes during the same time frame, and he is often credited with inventing the telescope. The historical records from the time are as fuzzy and imprecise as the images viewed through those early lenses. Wow, why are they so... They're being so rude. Don't make fun of their lenses. And any archived records have been lost over the centuries. We have an ad break. I'm going to finish this section while we're in the ad. By contrast, Van Leeuwenhoek and Hooke can thank ample documentation of their work for their respective legacies. Like Janssen, Van Leeuwenhoek began his work in obscurity, leaving behind few records. However, his friend, the prominent physician Rainier de Graaf, wrote a letter to the editor. Thank you, letter to the editor of the Philosophical Transactions of the Royal Society of London. My goodness, that is such a long title of a journal. Yeah, I'm gonna take a stretch during the ad break. Don't worry. Thank you, Ripco. I should put that as a channel point too, because I always forget. In his 1674, his report describing single-celled organisms produced controversy in the scientific community. But his observations were soon confirmed when the society sent a delegation to investigate his findings. He subsequently enjoyed considerable celebrity, at one point even entertaining a visit by the Tsar of Russia. Similarly, Robert Hooke had his observations using microscopes published by the Royal Society in a book called Micrographia in 1665. The book became a bestseller, a hit. The hit book Micrographia, and greatly increased interest in microscopy throughout much of Europe. I know they're probably talking about scientists in Europe were very interested, but it's funny to imagine this was a New York Times bestseller book about microscopes, and all of Europe became just obsessed with microscopy. Everyone, even the kids, the wives, the children, they wanted to know everything about microscopy. Zacharias Janssen. They may have invented the microscope. We'll never know. Okay, I'm gonna go to the bacterial room and do a little stretchy and maybe fill up my water. I'll be back. I'm back. I did a stretch and I got water. And I'm all good to go. I'm good to go now. Instruments of microscopy. Now we've learned how a microscope works. We've learned about the lenses. We learned about the history. Now it's time to learn about what the hell a microscope is. The early pioneers of microscopy opened a window into the invisible world of microorganisms, but microscopy continued to advance into the centuries that followed. In 1830, Joseph Jackson Lister, wow, what a name, created an essentially modern light microscope. The 20th century saw the development of microscopes that leverage non-visible light, such as fluorescence microscopy, which uses an ultraviolet light source, and electron microscopy, which uses short wavelength electron beams. These advances led to major improvements in magnification, resolution, and contrast. By comparison, the relatively rudimentary microscopes of van Leeuwenhoek and his contemporaries were far less powerful than even the most basic microscopes in use today. Damn. They're just dunking on them. It's okay, guys. I appreciate your rudimentary, basic, bad microscopes. In this section, we will survey the broad range of modern microscopic technology and common applications for each type of microscope. Light microscopy. Many types of microscopes fall under the category of light microscopes, which use light to visualize images. Examples of light microscopes include bright field microscopes, dark field microscopes, phase contrast microscopes, differential interference contrast microscopes, fluorescent microscopes, confocal scanning laser microscopes, and two-photon microscopes. Microscopes. That was a lot of times to say microscopes. Various types of light microscopes can be used to complement each other in diagnostics and research. I actually don't know most of these microscopes. I've never actually heard of them. I'm sure I've seen them, I just don't know their names, I guess. The bright field microscope, perhaps the most commonly used type of microscope, is a compound microscope with two or more lenses that produce a dark image on a bright background. Some bright field microscopes have monocular, having a single eyepiece, though most newer bright field microscopes are binocular, like the one shown in Figure 2.12. In either case, each eyepiece contains a lens called an ocular lens. The ocular lenses typically magnify images 10 times. At the other end of the body tube are a set of objective lenses on a rotating nose piece. The magnification of these objective lenses typically ranges from 4x to 100x, with a magnification for each lens designated on the metal casing of the lens. The ocular and objective lenses work together to create a magnified image. The total magnification is the product of the ocular magnification times the objective magnification. Oh yeah, I've definitely seen these. I just didn't know they're called a bright field. Or maybe I did. Oh, thank you for the raid, Myco Psycho. Existential snake raid. Thank you. Hello. We're just reading a microbiology textbook. We're reading it while I yap about what's going on. I hope you guys had a good time with Myco Psycho. Right up our alley, love it. We're learning a lot right now about microscopes. To be honest, I'm not the biggest fan of this section, but we gotta get through this all. We gotta be educated on all the aspects. Alright, so let's break down what a microscope is. There's the ocular lens piece, the objective lens, the stage, the diaphragm, the condenser, the illuminator. That's what it's called? A rheostat? Did anyone else know that? Or else was I just the type to just say that's like the light? That's the light adjusted guy. Oh, thank you for the follow, Myco Psycho. XY mechanical stage knobs. Okay, so why does the rheostat get a name, but this is called XY mechanical stage knobs? Fine focus, coarse focus, mechanical stage, revolving nose. Do you know how complicated it is to actually draw microscopes? I didn't realize this until I started drawing one myself, like for purposes of art, and I was like, this is so hard. There are so many pieces. The item being viewed is called a specimen. The specimen is placed on a glass slide, which is then clipped into place on the stage, a platform of the microscope. Once the slide is secured, the specimen on the slide is positioned over the light using the XY mechanical stage knobs. These knobs move the slide on the surface of the stage but do not raise or lower the stage. Once the specimen is centered over the light, the stage position can be raised or lowered to focus the image. The coarse focusing knob is used for large-scale movements with 4x and 10x objective lenses, with a fine focusing knob is used for small-scale movements, especially with 40x or 100x objective lenses. I really like something about the part where you like move the objective lenses on the nose piece. It's really satisfying. It really like, ooh, yeah. Except until like you have to be careful and it doesn't like it's not gonna crack anything. That's when it's scary. That's when it's not fun anymore. That's when you have to be, and you have to like put an oil, like you have to use oil. I don't like that. When images are magnified, they become dimmer because there is less light per unit of area. Highly magnified images produced by microscopes therefore require intense lighting. In a bright field microscope, this light is passed by an illuminator, which is typically a high-intensity bulb below the stage. Light from the illuminator passes up through the condenser lens, located below the stage, which focuses all the light rays on the specimen to maximize illumination. The position of the condenser can be optimized using the attached condenser focus knob once the optimal distance is established. Man, it's like I understand all this because when you use a microscope, these are just all the things. It's just crazy to think about. You can write paragraphs about how to do this. The condenser should not be moved to adjust the brightness. If less than maximal light levels are needed, the amount of light striking the specimen can be easily adjusted by opening or closing a diaphragm between the condenser and the specimen. In some cases, brightness can also be adjusted using the rheostat, a dimmer switch that controls the intensity of the illuminator. The fact it's called a rheostat and not just a dimmer is still crazy to me. A bright field microscope creates an image by directing light from the illuminator at the specimen. This light is differentially transmitted, absorbed, reflected, or refracted by different structures. Different colors can behave differently as they interact with chromophores, pigments that absorb and reflect particular wavelengths of light in parts of the specimen. Often chromophores are artificially added to the specimen using stains which serve to increase contrast and resolution. In general, structures in the specimen will appear darker to various extents than the bright background, creating maximally sharp images at magnifications up to about 1000x. That's huge. Bigly magnification. Further magnification, which would create a larger image but without increased resolution. This allows us to see objects as small as bacteria, which are visible at about 400x or so, but not smaller objects such as viruses. At very high magnifications, resolution can be compromised when light passes through the small amount of air between the specimen and lens. This is due to the large difference between refractive indices of air and glass. The air scatters the light rays before they can be focused by the lens. To solve this problem, a drop of oil can be used to fill the space between the specimen and an oil immersion lens, a special lens designed to be used with immersion oils. Oh yeah, this is type of like microscope work that always would like get me so nervous because I feel like it's so easy to break it. It's not really that easy to break it, but as like a student, I would be so nervous about it. I'd be like, oh my gosh, what if I didn't put enough oil? What if I put too much oil? Since oil has a refractive index very similar to that of glass, it increases the maximal angle at which light leaving the specimen can strike the lens. This increases the light collected and thus the resolution of the image. A variety of oils can be used for different types of light. Oh yeah, and cleaning it up afterwards. It's never actually like whoever used it last never did a good job cleaning it. It was always the case. I felt like somebody like it's always a little oily. Just a little greasy. Just break it on purpose. Okay, I've said that I've broken a centrifuge accidentally, but I feel like that led me to have a panic attack basically. And I think breaking a microscope would like literally have me like leave, like I would be gone. I would be gone. Yeah, put some olive oil on the microscope. It's like a, I don't know what kind of oil it is. I'm gonna, like, it was always in a special eyedropper bottle, but I've never actually looked at what oil they use. I'm guessing it's like mineral oil or something. That's not. Yeah, it was like a bench. It was a benchtop centrifuge. So it wasn't the end of. It wasn't the. It wasn't like a huge, like a huge one. But thankfully, no one was that mad. No one was that mad that it broke. It was an accident. It was a big accident. The rotor like broke apart. Don't ask me what I did. I can't even remember what I did because it was like I was like panicking so much. It's been blocked out of my brain. Microscope maintenance. Best practices. Just be a menace and break everything. No. Oh my gosh. It would be so scary when we would have labs in school, like in undergrad, and you have to like rent equipment from the school to be able to do some of these lab experiments. Especially like organic chemistry, we had to like rent, the different kind of like glasses that you need for experiments. And they would give us like all the glassware we need for a specific day in this case, and they would tell us like, if you break this, it's thousands of dollars. So don't break it. And I'd be like, okay, I won't break it. I promise I won't break it. Okay. And biggest I broke with a 4-liter beaker. Oh, you're much better than me. I think I've broken also so many like glass beakers. I'm unfortunately I was pretty clumsy when I worked in lab. It's also maybe another reason like experimental labs were never for me. My clumsiness is off the charts. I would drop things, or just it's like a little maybe too absent-minded. Even a very powerful microscope cannot deliver high-resolution images if it is not properly cleaned and maintained. Clean and maintain your things. The schools already ripped you off, yeah. I didn't want to. I was too scared. Yeah, when I like went into like a lab, like not that wasn't an undergrad like lab class, they were like, yeah, broken things can happen, just be honest when that happens and don't be too scared. I still didn't want to break anything though. And I'm guessing in undergrad, like for the classes, they're trying to be very careful because I'm sure if they say that, there's way too many people taking these classes, and there would probably be a lot of broken material, a lot of broken glass. They had to scare the students somehow into trying to be a little good, a little careful. Since lenses are carefully designed and manufactured to reflect light with a high degree of precision, even a slightly dirty or scratched lens will reflect light in unintended ways, degrading the image of the specimen. In addition, microscopes are rather delicate instruments, and great care must be taken to avoid damaging parts and surfaces. Among other things, proper care of a microscope includes the following. Okay, I feel like people sucked at taking care of microscopes. People were so bad. I never liked working with microscopes because they were always like filthy, or someone didn't turn it off. So many people would leave the microscopes on. It would be so frustrating that you come in and like you see someone just left their microscope just on, and crazy. Cleaning the lens with lens paper, not allowing lens to contact the slide. Protecting the bulb if there is one from breakage, not pushing an objective into a slide. Not using the coarse focusing knob when using the 40x or greater objective lenses. Only using immersion oil with a specialized oil objective, such as the 100x objective. Cleaning oil from immersion lenses after using the microscope. cleaning any oil accidentally transferred from other lenses. That's the one people would always forget. They would only clean the lens that they were like using at that time, but then they would forget that they used maybe another lens before, and so there's just oil on like so many different lenses. Covering the microscope or placing it in a cabin when not in use. Oh, it's okay. You can, even if you can't relate, I'm sure maybe it's relatable in one way when you see people not taking care of a communal good, and it's the most frustrating thing ever. Take care of the things that are for everyone to use, guys. Don't just leave it for the next person to take care of. Take care of it all of it. A dark field microscope. A dark field, the opposite of the bright field. Actually, no, it's not. A dark field microscope is a bright field microscope that has a small but significant modification to the condenser. A small opaque disc about 1 centimeter in diameter is placed between the illuminator and the condenser lens. This opaque light stop, as the disc is called, blocks most of the light from illuminator as it passes through the condenser on its way to the objective lens, producing a hollow cone of light that is focused on the specimen. The only light that reaches the objective is light that has been refracted or reflected by structures in the specimen. The resulting image typically shows bright objects on a dark background. So there's the lens, the light, blam, condenser lens. Ah. Dark field microscopy can often create high contrast, high resolution images of specimens without the use of stains, which is particularly useful for viewing live specimens that might be killed or otherwise compromised by the stains. For example, thin spirochetes like the Tryponium pallidum, the causative agent of syphilis, can be best used using the dark field microscope. Oh, this one. I see. I never knew it's called a dark field microscope. I always think about this as like, this is when you're like looking at things usually moving and wiggling around. Oh gosh, we're about to learn what happened to Cindy, guys. Okay, recap of part 1 of Cindy's story. Cindy is a camp counselor who scraped her leg at a camp, and it looks like there's a lot of pus, and then the camp nurse said we're gonna send it to get sampled because it doesn't look very good. Wound infections like Cindy's can be caused by many different types of bacteria, some of which can spread rapidly with serious complications. Identifying the specific cause is very important to select a medication that can kill or stop the growth of the bacteria. After calling a local doctor about Cindy's case, the camp nurse sends a sample from the wound to the closest medical laboratory. Unfortunately, since the camp is in a remote area, the nearest lab is small and poorly equipped. A more modern lab would likely use other methods to culture, grow, and identify the bacteria, but in this case, the technician decides to make a wet mount from the specimen and view it under a bright field microscope. In a wet mount, a small drop of water is added to the slide and a cover slip is placed over the specimen to keep it in place before it is positioned under the objective lens. Under the bright field microscope, the technician can barely see the bacterial cells because they are nearly transparent against the bright background. To increase contrast, the technician inserts an opaque light above the illuminator. The resulting dark field image clearly shows that the bacteria cells are spherical and grouped in clusters like grapes. Oh god. Oh god, is Cindy gonna be okay? We gotta keep going. We have to trudge. Yeah, she has staff. She definitely does. We have to find out if she's okay, though. And we have to trudge through understanding we have to learn more about microscopes before we can figure out if she's okay. You never know with these clinical focuses. They might have like a crazy turn of events. Phase contrast microscopes use refraction and interference caused by structures in a specimen to create high contrast and high resolution images without staining. It is the oldest and simplest type of microscope that creates an image by altering the wavelengths of light rays passing through the specimen. To create altered wavelength paths, an annular stop is used in the condenser. The annular stop produces a hollow cone of light that is focused on the specimen before reaching the objective lens. The objective lens contains a phase plate containing a phase ring. As a result, light traveling directly from the illuminator passes through the phase ring, while light refracted or reflected by the specimen passes through the plate. This causes waves traveling through the ring to be about 1/2 of a wavelength out of phase with the passing through the plate. Because waves have peaks and troughs, they can add together if in a phase together or cancel out each other if they're out of phase. When the wavelengths are out of phase, wave troughs will cancel out wave peaks, which is called destructive interference. Structures that refract light then appear against a bright background of only unrefracted light. More generally, structures that differ in features such as refractive index will differ in levels of darkness. Yeah, there's lots of history in this book. I kind of like the history lessons are kind of interesting. It's always funny to see how people like in the 1600s were doing some crazy big brain shenanigans, and nobody like gives them like they didn't even know they were right until centuries later. Whoa. I feel like there's so many levels of light here. Illuminating light, diffracted light, undiffracted light. And then the combined light over here. This diagram. Yeah, you go. I remember everything about the guy who called bacteria wee little beasties and animalcules, but I don't know anything about what a bacteria is. Please send help. Because it increases contrast without requiring strains, phase contrast microscopy is often used to observe live specimens. Certain structures such as organelles in eukaryotic cells and endospores in prokaryotic cells are especially well visualized with phase contrast microscopy. Oh, look how good it looks out here. You can see all the doodads. Barely visible. Look at all the little details. Wow, it looks all chunky. I like it. Differential interference contrast microscopes. DIC microscopes. Also known as Nomarsky optics. Yeah, it's such a crazy improvement. Like, the fact that, like, it looks relatively flat into, like, oh, you can tell how it's got, like, depth to it. So cool. Are similar to phase contrast microscopes in that they use interference patterns to enhance contrast between different features of a specimen. In a DIC microscope, two beams of light are created in which the direction of wave movement polarization differs. Once the beam passes through either the specimen or specimen-free space, they are combined, and effects of the specimen causes differences in the interference patterns generated by combining of the beams. This results in high contrast images of living organisms with a three-dimensional appearance. These microscopes are especially useful in distinguishing structures within live, unstained specimens. Wow. That's so cool looking. Leonian Fonsesia pedrosoi, grown on modified Leonian's agar. What a funky little fungus guy. It looks like it's got like I like how it's like so tree-like. It looks like a tree like I would have drawn as a kid. That's so cool. Fluorescence microscope. Woo, this is like such a big part of science, fluorescence microscopy. A fluorescence microscope uses fluorescent chromophores called fluorochromes, which are capable of absorbing energy from a light source and then emitting this energy as visible light. Fluorochromes include naturally fluorescent substances such as chlorophylls, as well as fluorescent stains that are included in the specimen to create contrast. Living organism in 3D images. Is this how Nico gets recorded to Yes, you're exactly right. You're right. I've had like fun ideas because in the future I would love to get a 3D model, and I've had some very fun ideas of like how I would want to set up that stream that involve funny little microscope, like a funny little microscope skit. Maybe one day we will be able to do that. I have to first learn. I have to first learn finish my lessons on using 3D programming. Fluorochromes include naturally fluorescent substances such as chlorophylls, as well as fluorescent stains that are added to the specimen to create contrast. Dyes such as Texas Red and Fitzy are examples of fluorochromes. Other examples include the nucleic acid dyes. You're not gonna make me say DAPI, but and acridine orange. Acridine. The microscope transmits an excitation light, generally a form of EMR with a short wavelength such as UV and blue light, toward the specimen. The chromophores absorb the excitation light and emit visible light with longer wavelength. The excitation light is then filtered out, in part because UV light is harmful to the eyes. Okay, that's true. That's true, parentheses, that's true. Yeah, it would be a fun animation. God, there's so many things to do, and then I complain that I don't have time to do it, and then I spend 3 hours, almost 4 hours reading a textbook. So that only visible light passes through the ocular lens. This produces an image of the specimen in bright colors against a dark background. Fluorescence microscopes are especially useful in clinical microbiology. They can be used to identify pathogens, to find particular species within an environment, or to find the locations of particular molecules and structures within a cell. Approaches have also been developed to distinguish living from dead cells using fluorescence microscopy based on whether they take up particular fluorochromes. I am having a lot of fun. It's just kind of fun. It's actually really fun to learn this stuff. It's not new things, but it's just fun to recap. And also just yap to you guys. Sometimes multiple fluorochromes are used in the same specimen to show different structures or features. One of the most important applications of fluorescence microscopy is a technique called immunofluorescence, which is used to identify certain disease-causing microbes by observing whether antibodies bind to them. Oh boy, immunofluorescence. Antibodies... Thank you, parentheses. It's probably a good idea to explain what an antibody is, because they have not introduced them yet. This is a foreign concept. If this is like you're completely new to microbiology and you're just reading like we are from like start to finish. Antibodies are protein molecules produced by the immune system that attach to specific pathogens to kill or inhibit them. There are two approaches to this technique: direct immunofluorescence assay, DFA, and indirect immunofluorescence assay, IFA. In DFA, specific antibodies, those that target the rabies virus, are stained with a fluorochrome. If the specimen contains the targeted pathogen, one can observe the antibodies binding to the pathogen under the fluorescent microscope. This is called the primary antibody stain because the stain antibodies attach directly to the pathogen. Oh. I feel like we typically use not this. Or at least yeah. In IFA, secondary antibodies are attached with a fluorochrome rather than primary antibodies. Secondary antibodies do not attach directly to the pathogen, but they do bind to primary antibodies. When the unstained primary antibodies bind to the pathogen, the fluorescent secondary antibodies can be observed binding to the primary antibody. Thus, the secondary antibodies are attached indirectly to the pathogen. Since multiple secondary antibodies can often attach to a primary antibody, IFA increases the number of fluorescent antibodies attached to the specimen, making it easier to visualize the features in specimen. Oh yeah. So, direct is guy attaches right to the guy. Fluorescent guy attaches right to the antigen. Indirect, fluorescent guy attaches to a guy that's attached to the guy that you're looking at. There's layers to it. I think like this is like typically what we did for like Western blots. Like you would have your proteins on your membrane, and then you wash it with one like primary to bind to what you're looking for, and then you wash it again with the secondary antibody to bind to what those antibodies you first put in. It makes sense why it's easier this way. Because trying to bind directly to the antigen can be probably pretty difficult, especially if you're not sure exactly what you're looking for. Confocal microscopy. Whereas other forms of light microscopy create an image that is maximally focused at a single distance from the observer, A confocal microscope uses a laser to scan multiple Z-planes successfully. I never did confocal microscopy, but I know that this was like really expensive to book this experiment, like at least in my school. And I remember it would always be packed. It would be really hard to get time to use the confocal microscopes. This produces numerous two-dimensional high-resolution images at various depths, which can be constructed into a three-dimensional image by a computer. Like, I can understand just reading that sentence is kind of like this would like explode the brains of all those historians that we read about. They took many two-dimensional pictures at different depths and they make 3D image. And blotting being told now it's running. Yeah, I hated Western blotting. It would be, hey, yeah, it's just a lot of waiting time, and then you have to go analyze the pictures. So this obviously depends on like your lab, but for me, the lab I was in, when we would do Western blots, we didn't have like a machine, like those machines to easily read what your Western blot results were. We would like go into a dark room, put a sheet of X-ray paper, like so that the fluorescence would mark the X-ray paper, and then I'd have to like make sure to do all of that, like do all the sensitive procedures in this dark room where I can't see anything. And I'm just like feeling around like with my hands to go, okay, there's the table, there's my membrane, there's the X-ray paper. I have to put this on carefully. It was such a nightmare. I was like so jealous of labs that had like a that could easily read their Western blots without doing all this darkroom nonsense. Confocal microscopes are thus very useful for examining thick specimens such as biofilms, which can be examined alive and unfixed. I vaguely remember, yeah. Western blotting is such like it's also just like a multi-step process, and there's a lot of waiting in at every stage of it. There's a lot of waiting. Look how cool that is. It's so funky looking. I like it. 2 photon microscopes. While the original fluorescent and confocal microscopes allowed better visualization of unique features in specimens, there were still problems that prevented optimum visualization. The effective sensitivity of fluorescence microscopy when viewing thick specimens was generally limited by out-of-focus flare, which resulted in poor resolution. This limitation was greatly reduced in confocal microscope through the use of a confocal pinhole to reject out-of-focus background fluorescence with thin, less than 1 micrometer, unblurred optical sections. However, even the confocal microscopes lacked the resolution needed for viewing thick tissue samples. These problems were resolved with the development of the 2-photon microscope, which uses a scanning technique, fluorochromes, and long wavelength light to visualize specimens. Holy cow, I feel like these microscopes they must be expensive. The low energy associated with the long wavelength light means that 2 photons must strike a location at the same time to excite the fluorochrome. The low energy of the excitation light is less damaging to cells, and the long wavelength of the excitation light more easily penetrates deep into thick specimens. This makes the 2-photon microscope useful for examining living cells within intact tissues. Man, brain slices, embryos, whole organs, and even entire animals. What the frick? I feel like aren't these microscopes just the best? Yeah, okay. I figured that this is going to talk about the cost. Currently, use of 2-photon microscopes is limited to advanced clinical and research laboratories because of the high cost of the instruments. A single 2-photon microscope typically costs between 300 grand and 500 grand, and lasers used to excite the dyes used on specimens are also very expensive. However, as technology improves, 2-photon microscopes may become more readily available in clinical settings. Holy cow! They don't even have a picture. I can't even get to visualize what like 500,000 microscope produces. They don't even give us an example. It's that just that expensive. Man. Electron microscopy. The maximum theoretical resolution of images. This microscope costs more than my home. Yeah, I mean, like, microscopes are very expensive to begin with. Like, they're pretty expensive for, you know, how generally they're used, but $500,000 is insane. I'm wondering if these are big. Kinda curious. 2-photon microscope. Are they huge or something? Not really. They look like just like your average microscope, which is funny. Example. There's not really many example pictures. I guess that makes sense. Okay. Most light microscopes can only magnify 1000x, and a few can magnify up to 1500x, but this does not begin to approach the magnifying power of an electron microscope. EM, which uses short wavelength electron beams rather than light to increase magnification and resolution. Electrons, like electromagnetic radiation, can behave as waves, both wavelength of 0.005 nanometers. They can produce much better resolution than visible light. And EM can produce a sharp image that is magnified up to 100,000x. Though EMs can resolve subcellular structures as well as some molecular structures, e.g. single strands of DNA. However, electron microscopy cannot be used on living material because of the methods needed to prepare the specimens. Dead only. Not living. No living. There are two types. I will hydrate. Thank you. There are two types of EM, the transmission electron microscope, TEM, and the scanning electron microscope, SEM. The TEM is somewhat analogous to the bright field light microscope in terms of the way it functions. However, it uses an electron beam from above the specimen that is focused using a magnetic lens rather than a glass lens and projected through the specimen onto a detector. Electrons pass through the specimen and then the detector captures the image. Oh, it's all funky looking. I know what an electron microscope is, but I've actually never seen one. It's kind of cool. Look at this old, like, ignore the microscope. Look at this keyboard and mouse and these monitors. Like, isn't that that's just how science is. You have your, like, they give you this keyboard. That's in every lab I've been in. This dirty black keyboard. It's like, you're supposed to take off your gloves usually when you touch any, like, you know, computer equipment, but it's like it's so nasty, the keyboard, that you want to keep your gloves on every time. Oh yeah, for sure. It's legacy hardware support. Woo! And if it breaks, like, if you don't understand, like, if it somehow stops connecting to the computer, you will never be able to figure out what broke and how to fix it until you call this company, and they send, like, some guy that's like 7 years old and he's been doing this his whole life. And they will never upgrade it. They will never update the monitors or PC. TEM, the electron gun. Boom, 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 and electrons. For electrons to pass through the specimen in a TEM, the specimen must be extremely thin, 20 to 100 nanometers thick. That's thin. Tiny, tiny, tiny. You don't get new parts from a manufacturer. You get it from a lucky... Yeah, you ask the manufacturer, hey, like, this broke. Is there any way we can get a new thing? And they go, we haven't produced any of these since, like, the 80s. You might want to go ask some other labs if they have, like, a spare part. The image is produced because of varying opacity in various parts of the specimen. This opacity can be enhanced by staining the specimen with materials such as heavy metals, which are electron dense. TEM requires that the beam and specimen be in a vacuum and that the specimen be very thin and dehydrated. The specific steps needed to prepare a specimen for observation under an EEM are discussed in detail in the next section. SEMs form images of surfaces of specimen, usually from electrons that are knocked off of specimens by a beam of electrons. This can create highly detailed images with three-dimensional appearance that are displayed on a monitor. Typically, specimens are dried and prepared with fixatives that reduce artifacts, such as shriveling. That can be produced by drying before being sputter-coated. I like that word, sputter-coated. Sputter-coated. With a thin layer of metal such as gold, whereas transmission electron microscopy requires very thin sections and allows one to see internal structures such as organelles and the interior of membranes. Scanning electron microscopy can be used to view the surfaces of larger objects, such as pollen grain, as well as surfaces of very small samples. Some EMs can magnify an image up to 2 million x. Wow. I've never seen magnification that kind of magnification. To expand both the resolution and type of materials that can be examined, Pratibha L. Guy and Edward D. Boys invented the Environmental Transmission Electron Microscope, the ETEM. The device advanced the standard electron microscope by incorporating a gas-filled chamber in which a wider array of substances could be contained. Thus, researchers could observe actual reactions taking place at incredible magnifications. Guy later improved on her work by creating the Environmental Scanning Transmission Electron Microscope, the ESTEM, which has even greater resolving power. Guy herself was the first person to ever see individual atoms interacting. Wow, what a career. You not only you help invent one electron microscope, you go on to invent a better electron microscope than the first one, and then you see atoms interact. What a amazing lady. Yeah, I want to see that particular. Yeah, I would like to view nitrogen. The TEM image of cells in a biofilm shows well-defined internal structures of the cells because of varying levels of opacity in the specimen. That's so cool. Look, you can really see the organ, like you can see it's like stuff. That's cool. This color-enhanced SEM image of this bacterium Staphylococcus aureus illustrates the ability of scanning electron microscopy. I think, isn't that insane that you're able to see it at this level of magnification? Like, this blows my brain. Because this looks like almost like a graphical image. And when I say graphical image, it sounds stupid. But I mean, it looks like someone, I could have just, drawn this. But it's, the microscope can show that. That's so cool. It looks like grapes. You could also tell me that's grapes. Using microscopy to study biofilms. Oh, I love biofilms. A biofilm is a complex community of one or more microorganism species, typically forming as a slimy coating attached to a surface because of production of an extrapolymeric substance, EPS, that attaches to a surface or at the interface between surfaces, e.g. between air and water. In nature, biofilms are abundant and frequently occupy complex niches within ecosystems. Biofilms are, I would say maybe biofilms are more interesting. They're so complicated though. They can add a lot of complexity to what you'd think would be something simple to study and research. Especially when it comes to medicine. In medicine, biofilms can coat medical devices and exist within the body. Because they possess unique characteristics such as increased resistance against the immune system and to antimicrobial drugs, biofilms are of particular interest to microbiologists and clinicians alike. Because biofilms are thick, they cannot be observed very well using light microscopy. Slicing a biofilm to create a thinner specimen might kill or disturb the microbial community. You slice them and then they just die. They go, eh. Confocal microscopy provides clearer images of biofilms because it can focus on one Z-plane at a time and produce a three-dimensional image of a thick specimen. Fluorescent dyes can be helpful in identifying cells within the matrix. Additionally, techniques such as immunofluorescence and fluorescence in situ hybridization, fish. I like that acronym, fish. I don't know much about this technique, except the fact that when I do my job with cancer therapeutics, a lot of times you see fish as like an important way to identify that they did have a biomarker present, especially things like the gene ERB2, like amplification. It's usually confirmed via fish. So I'll like write fish. In which fluorescent probes are used to bind to DNA can be used. Electron microscopy can be used to observe biofilms, but only after dehydrating the specimen, which produces undesirable artifacts and distorts the specimen. In addition to these approaches, it is possible to follow water currents through the shapes, such as cones and mushrooms of biofilms, using a video of movement of fluorescently coded beads. Ooh. Diagram showing 5 stages of biofilm development of I'm so bad at pronouncing that one. It trips my tongue up. Stage 1. Look, it attached. Irreversible attachment. Maturation. 2. Dispersion. Whoa. That's so cool. Multiple species of bacteria grow in a biofilm on a stainless steel. Yeah, I'm not a fan of that one. Scanning probe microscopy. Okay, I'd never heard of this one. A scanning probe microscope does not use light or electrons, but rather very sharp probes that are passed over the surface of the specimen and interact with it directly. This produces information that can be assembled into images with magnifications up to 100 million x. Okay, at a certain point, like, I feel like we need to, like, simplify these numbers. Such large magnifications can be used to observe individual atoms on surfaces. To date, these techniques have been used primarily for research rather than diagnostics. There are 2 types of scanning probe microscopes: the scanning tunnel microscope, the STM, and the atomic force microscope, the AFM. An STM uses a probe that is passed just above the specimen as a constant voltage bias creates the potential for an electric current between the probe and the specimen. So you have the thing and then what you're looking at. And then it has just constant voltage. This current occurs via quantum tunneling of electrons. Oh my gosh. Okay, this is some high tech. This is some high, like, physics techniques over here. Yeah, it's just literally poking it and then sending voltage. It's just very sciencey poking is a good way to put it. This probe is moved horizontally above the surface and the intensity of the current is measured. Scanning tunneling microscopy can effectively map the structure of surfaces at a resolution at which individual atoms can be detected. Similar to an STM, AFMs have a thin probe that is passed just above the specimen. Oh, okay, so the one is poking it and one is going, I'm not touching you. I'm not touching you. However, rather than measuring variations in the current at a constant height above the specimen, an AFM establishes a constant current and measures variation in the height of the probe as it passes over the specimen. As the probe tip is passed over the specimen, forces between the atoms, the van der Waals force, capillary force, chemical bonding, electrostatic forces, and others cause it to move up and down. The deflection of the probe tip is determined and measured using Hooke's law of elasticity, and this information is used to construct images of the surface of the specimen with resolution at the atomic level. Wow. I feel like it's crazy. I feel like these are very different. Despite both being part of the scanning probe microscopy, they definitely are giving very different feels. Yeah, he's just like going, do something. Do something. I like how this one looks, the AFM one. It has like it looks really just weird. I don't really understand what's going in the STM one, but I guess those are just atoms. But my brain can't comprehend that these are individual atoms. And this is just a review of all the microscopes we saw. There's so many. Staining microscopic specimens. By the end of the section, you will be able to differentiate between simple and differential stains. How long is this section? I'm just curious. Okay. I just wanted to see how much longer we had. I think this chapter is pretty long. But we might have to call it here. Considering I feel like I've been yapping and reading to you guys for 4 hours. Okay, I'm gonna start the ad break and get some water in the meantime. I'll be right back. Hi, I got water. I do think I have to end it before I've ruined my throat for reading too much. It's already been like 4 hours of reading. I want to try. I might do this like every Friday until we get through it or I get bored of it. But so far I've been having a lot of fun, so I think I might make this my science friyay weekly special. And eventually we'll get through all 1,221 pages. We actually made really good progress today. We're on page 71. Page 71 is kind of crazy. And it feels like it flew by. I can't wait, I'm spoiling it. Don't look. So I think we will be calling it to both the progress and reoccurring subject. Yeah, I feel like people seem to be enjoying this, so I will definitely do this. And I'm excited to keep reading it. And maybe whenever we get through this textbook, we can get another textbook going. It's pretty fun and it's relaxing. So I think I wonder if I can find someone to raid. And it's suggesting I don't know anybody in this category. And I need to tweet about who to raid. I have to find I have to find a list of people I'm okay with raiding. Cause I always get stun locked trying to figure out who to go after. Anyway, I'll do that tomorrow. I will be here tomorrow. It's gonna be a co-working stream in the afternoon. I have a lot of things to catch up on, and we'll do some. We'll work together while I yap. You won't be able to see what I'm working on because I can't show you, but I'll be here yapping while I work, and maybe you can get some work done too. Work? No, no, no. Work good. Or you don't have to work. You can watch. You can listen to me work. You know. It'll most likely be. Okay, you don't have to work. You don't have to work. You don't have to work. You can watch. You can suffer having somebody else work. You know, you can watch me work because I have a lot of work to do. It's mostly fun work, but I just have a lot of things to go through. I've been ignoring. Anyway, we're gonna call it here. I'm gonna rest my throat and like be a potato for the rest of the evening. I will see you guys later. Thank you for hanging out and listening to me read a book for 4 hours. Okay. Bye 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 bye.